Det er en test. Hører dere meg?
All right, thank you. Welcome to everyone. It's fantastic that so many, almost 150 persons, have signed up for this conference. I hope you all make yourself comfortable. Whatever you are here at Nina House, at your office, or in your own home. This year, Living Norway Colloquium is called Towards Openness and Transparency in Applied Ecology. My name is Norunn Myklebust, and I am the director of Norwegian Institute for Nature Research, NINA. We are an independent foundation focusing on environmental applied research, emphasizing the interactions between human society, natural resources, and biodiversity. Many of our research areas are associated with natural resource conflicts. Examples of such areas are impacts and mitigation in renewable energy, like hydropower and wind energy, aquaculture and conflicts between farmed and wild salmon, and conflicts between carnivores and farmers. The conflicts may have several reasons, and many of them is, of course, very difficult and may be impossible to solve completely. However, a common knowledge base is always an advantage. To create a common knowledge base, transparency, openness, and of the data and the methods is essential. I'm very happy that NINA and our Living Norway partners are taking lead in this field. I also want to thank the Norwegian Research Council for supporting this conference. It's an exciting program with both the lecture on key topics and workshops. I hope all participants are ready to join and discuss wherever you are sitting and follow the conference. I hope we can create and reach open and reproducible science for the 21st century. So welcome to all and good luck. Thank you. And I will give the floor to our partner from Artsdatabanken. Please, the floor is yours.
I have to do that. Thank you. Yes, my name is Wouter Koch. I am a PhD student at NTNU, also a senior advisor at the Artsdatabanken. And I will be the chair for the first session today. So what I would like to do is just go through the program, which should be up here somewhere. There we go. Yeah, this is a great place to start for anyone attending this conference. livingnorway.no. You will find all of the information under the conference page for 2020. The program for today. There we go. This is going to be a bit tiny, isn't it? There we go. So I'm going to have 5 minutes of just a short house. After that, Ireland will have the floor, talking about the Living Norway Ecological Data Network, After that, we will have Anders Finstad about data standards. We are honored to have Dag Andresen from GBIF with us to talk about GBIF, I guess. We have Matthew Granger talking about the data management with Living Norway R package. And then we will close this session with Caitlin Mandeville, who talk about the data from a user's perspective. After that, we will have lunch. So we're going to do this whole thing on Slack, whether you're physically here or out there in the world. We're all going to be using Slack so we can have all of the questions gathered one place, and the presenters will have the opportunity to go in later and answer all of the questions. A great way to do that is when you see a question that you support, give it a little thumbs up or unicorn or whatever you want to use, so that we as moderators can see, okay, this is a question that several people have. For those of you who are not familiar with Slack yet, you all got an invite per email. Slack's pretty easy. You go on slack.com, you log on with the credentials that you got, and you get this, you get a screen like this. There's a mobile app as well. There's a desktop app. I'm just showing the website here. So all of this is the workspace. We have one workspace for the whole conference. The workspace has several channels, which are the sessions, announcements, the live stream. We have some welcome channel. So these are kind of the topics that we are going to be talking about. And then we have, in each session, we have pre-made threads where you can ask your questions. So if you have a question for Ireland, there's a thread here, Living Norway Ecological Data Network, where you can go in and reply. Here you can ask your questions. If you have any trouble using this at all, get in touch with someone from the organization and we'll help you. And if you get to the point where you're already on Slack, you can also ask your questions on Slack itself, and we'll make sure to help you. There's a code of conduct. I won't go too much into that. It's pretty basic. Just be nice to each other, whether you're online or offline. And we have Sam, who will be enforcing these rules with a strict but fair hand. Yeah, I think we won't be needing Sam to do that much, I hope. There's a survey that has been circulated. Please all answer that. These things are important for us. A few practicalities for those physically here. Nearest emergency exit is right behind the curtain on that side. If it happens to be a fire right over there, I suggest we go up the stairs, I guess. Other than that, there's lunch. Please be on time. Try to keep your time, as I'm doing. There will be a very quick break between each of the presenters. We'll try to keep the program, and if you tweet about this event, please use the LN2020 hashtag so we can find it. I think that's it for me. Ireland, are you ready? Then I would like to give the floor to Ireland. I guess this is yours? Yep.
This is not mine, no.
Okay. Well, you find it.
stikkordet, Anette. Jeg vet ikke hvor presentasjonen min er. Thank you. Yeah. Okay. I'm on, yeah? Yes. Strictly speaking, we are a couple of minutes before the program. So, so I can talk a little bit before I will start with my lecture. So, first of all, you have already been wished welcome by Norun Myklebust, the director of NINA. So I will also wish you all welcome on behalf of the Living Norway Ecological Data Network. One thing that Walter mentioned briefly was that we have a survey. We circulated the survey to all of the participants. All of you that have signed in received an email. Actually, you have received 3 emails now. We will really encourage you all to fill in this form. This is the first part of a survey with 3 parts, and this first part will be possible to fill in until after lunch today. You received the last email this morning, and we really hope that you will fill in this. For us, it's important to develop the program and to plan for the future, and we are also planning to use the responses for a scientific publication on these issues. So please, if you haven't done so far, quite a few of you have done so, but those of you that didn't fill in this yet, please do so. And yeah, you can do so in the breaks, and there will be time in the lunch from 12 o'clock to 1 o'clock. After that, sometime after that, we will close the first part, and we will get ready for the second part, which you will receive a mail about later. So yeah, I think we are getting close now to the real presentation. So again, it's a pleasure to welcome you all to this conference. As has been mentioned already, we are close to 150 participants, which is a really high number, and I must admit a higher number than what we expected when we started to plan this conference. It's great that we can have events like this these days, and that we can meet online and make these events even with the current global situation. My name is Erlend Nilsen. I'm a project leader of the Living Norway Ecological Data Network. And in this first session now, we will have several presentations from friends and colleagues from the network that will present much more in detail what we are planning to do, what are our goals, our visions, and the more technical ways that we see this is moving forward. So for me, I will just give a more of a general introduction to the topic, and if you think of this as a paper, this will sort of be the introduction to the paper, and then the others will present the methods and the results and discussions and so on. We are living in a time where science is probably stronger tied to political developments than ever before, I think. And this was discussed in the last week's editorial in Nature. This applies both in terms of how funding and research policy priorities are going and conducted. I think that the global coronavirus pandemic has highlighted this, how interdependent these two enterprises are. And it's also clear that sometimes politicians have the power to overlook and neglect scientific evidence when making decisions. And we have seen that how the political situation some places in the world are making it tricky to come across with scientific findings and so on. So I think in these times, openness around the processes that we as scientists are part of when we are conducting science is more important than ever, that we are open around these processes. As you all know, we live in a world where the landscapes, the biosphere, the atmosphere, and so on, are heavily affected by human activities. And this is commonly now and more or less accepted, that this represents a new epoch, starting sometimes in the 40s, 50s, 60s. And this is our epoch, where the biosphere is strongly modified and affected by human activities. So this affects the way we are doing applied ecology or ecology these days. Of course, Charles Darwin, 160 years ago roughly, when he wrote and published his book about the origin of the species, he described how the tree of life had evolved over long geological timescales and resulting in the species that we can observe today. And many of the species we haven't even observed and described. But we also know that much of this tree of life, the distribution, the abundance, and existence of species is changing. It's under threat from human activities and anthropogenic pressures. Many of you will recognize the figure to the left as coming from the summary report from the last year's report from the Intergovernmental Science Policy Platform on Biodiversity and Ecosystem Services, the IPBES panel, which describes how the different biomes have been affected by various human activities. The figure to the right is from the relatively recent Living Planet report that came out this year with the title Bending the Curve of Biodiversity Loss. And this one depicts the Global Living Planet Index from 1970 up until 2016 and shows that for the species populations that are included in this index, the mean abundance has decreased with roughly 68% over this time period. So this is part of doing applied ecology these days. This is also part of doing applied ecology these days, that the World Economic Forum has identified the biodiversity crisis and the loss of biodiversity among the top 5 risk factors for the next 10 years. So what we are, when we are working as ecologists these days, of course many of us got into this from a fundamental interest for the nature and processes and natural history reasons and so on, but it has really a fundamental societal value, the work and the research we are doing. And just to reiterate, I mean, ecology, it has many definitions. One of me as a background as a vertebrate biologist, working mainly with mammals, birds, population ecology, population dynamics, I like this definition of ecology, which says that ecology is the scientific study of the interactions that determine the distribution and abundance of organisms. This goes back to Charlie Krebs, 1972. And there are many other definitions also. But this one tells us that ecology is about understanding the processes that So, if you think back to the 2 former slides, this puts ecology and applied ecology really at the center stage of the societal challenges these days. We need to understand the processes and the interactions that determine the distribution and abundance of the organisms that live on the planet. Ecological research takes place in a lot of different spatial scales. A lot of the research that is reported in papers and in reports and so on takes place on a relatively small scale. It happens on local scales or on a landscape scale. Also, most of the research that we report is observational. It's not based on experiments. So this is the data at hand when we are trying to solve the big questions and big challenges. And of course, it's a lot of value in the small scale work. I've been doing that myself in many cases, and there is a lot of value in that. But sometimes we need to also be able to scale up, to understand the processes at larger scales. In many cases, this means that we need to integrate and merge data from several disparate data collection processes, from different research programs, from different monitoring programs. And these days, the statistical machinery to do so is developing quickly. There's a lot of research now going into data integration models, statistical data integration, integrated population models, integrated species distribution models, and so on. And the cornerstone here is to use data from different programs, from different data sets, integrate them into common statistical framework, and get more out of the data at hand, more than you can learn from separate analysis of the separate data sets. To make this efficient on a larger scale than it's currently done, we need to mobilize, make available a much larger body of the data that is actually collected and is available out there. This small example here just show how, for instance, in Norway, you have two different sources of large-scale data for willow ptarmigan abundance distribution. There is a line transect distance sampling survey data on the left side of the country. And then there is occurrence data from citizen science projects. These are different data sources. They can, and we are doing so, integrating them in common statistical frameworks. But to do so efficiently and for more cases, we need to have them interoperable in the same formats, and they need to be available for fellow scientists. Yeah, so open science in applied ecology, open science and open data. We will hear much more about open science later today. We'll have a couple of talks specifically on this. But in general, again, there are many definitions. But in general, open science can be defined as a movement to make scientific research, data, and dissemination accessible to all levels of an inquiring society. So it means you're opening up the process of doing research, opening up data, opening up the results published in papers, opening up access to your computer code, and so on and so forth. And one of the things that open science can do is to help us reduce research waste. It can help us target the correct questions, and it can help us in a much better way delivering scientific results that can be used by society at large. I will not go through all the details here. It was summarized in a paper we had this year with Matt Granger, which we'll present later today. the first author on, where we reported how open science, open data can help us reduce research waste at several levels. And of course, open data is a central part of open science. By making the research data and monitoring data available to the society, there is a lot of benefits to that. It includes things like verification of published results. It's hard to verify published results if your fellows cannot access your data and your computer code. It creates the foundation for better meta-analysis that we will hear more about later today. It provides possibilities to test new questions that are not necessarily put forward by the PI and the person that collected the data. It has the potential to increase citation rates and credit to those that provide data if it's done in a correct and a good way. It provides new teaching opportunities. It reduces the risk of data loss. All of these were included in the important and nice paper by Whitlock in 2011, and I've added a couple of more here. It makes it possible to integrate data across larger spatio-temporal domains, which is very important these days when we are doing these large-scale assessments. It increases the efficiency. We are spending these days a lot of time to find and compile and set together ecological data in all research projects, and the next research project typically do the same. And it should increase trust and transparency in the process. And of course, everybody is expecting open data these days. Public bodies write about this, research funders do so, journals all expect open data, which is good. But the current practice is very, very fragmented. There are few clear guidelines on how we do this. People are doing it in very different ways, sometimes in efficient ways that makes the data reusable by others, and sometimes in less efficient ways. So this is what many fellow researchers, many ecologists meet. They support the idea, the principles, but basically a lot of people don't know exactly how we do this in the best way. And there has been some surveys of the current situation. How well are we doing when it comes to making code data available, and how reusable are the data and the code that are there? We will hear more about this just after the lunch, but here are 3 examples of paper that do touch upon this question. And in general, I think the conclusion is that there is scope for improvement, without doubt. Fair data is a concept that many of you will be familiar with. This will again be discussed, presented more in detail later today. It basically means that when you publish, archive your data, they should be findable, accessible, interoperable, and reusable. There are some clear technical guidelines here, but I think one thing that is often forgotten is that to do this in the best possible way, it also means that you should, for your data, as long as possible, use standards, established standards, open standards, to make them more interoperable and more reusable by your fellow researchers and the society as such. Anders Finstad will talk more about this after, but this is a very important part if you want to make the most out of the process. Yeah, there has been some surveys, quite a few in different contexts, about what people, how researchers report or find obstacles to public data sharing. And this one specific one is conducted by Springer Nature, published in 2018. And in this case, the researcher that responded to this pointed to three to four main obstacles. One was how to organize the data in a presentable and usable way. So a lot of researchers don't know exactly how to do this, which is not surprising. Not knowing which repository to use is another central one. Unsure about copyrights and licensing issues is another central one. And lack of time to deposit data. We will touch upon many of these in the talks later today and how we can reduce these barriers. So, tuning into what we are up to in the Living Norway Ecological Data Network. We will have a couple of more talks before lunch now about more detailed plans and visions and technicalities about how we hope to help on this situation. But the main objective of our network and the infrastructure we are building is to make accessible, standardized and documented data from field-based ecological research and monitoring. And then we think that we need to do a lot of different activities to achieve this. Currently, the network consists of 8 Norwegian institutes that are listed here. And we should point out that we are tightly connected to the Global Biodiversity Information Facility, GBIF. We build on the same standards, which Anders will talk more about, Anders Finstad will talk more about later, and we build on the same underlying infrastructure for making data available. So in a way, we are trying to expand the focus and the applicability of the Darwin Core Standard and this data flow to cover also ecological data to a larger extent. We think that we need to do so, well, by focusing on data harmonization, standardization for ecological data. We will do so by contribute to the international, and we will have a lot of focus on education and training of students and researchers in modern data management and ethics related to use and provision of open various national and international frameworks. So to sum up, before you give much more details about what we are up to, I will just take the quick summary. We see that the need for ecological research is real, global, and unquestionable. We see that the scale of the challenges we are up to in society requires large-scale collaborations, efficient use of resources, and research practices that build trust around the research and the evidence base. And basically, the Living Norway Ecological Data Network is our response to many of these challenges. And I would like to end with a small note that targeted actions can help. This paper was published last year by Frederikke Bolland and co-workers, and they used expert elicitation to see how many species during since 1993 roughly has been birds and mammals was saved from extinction by conservation actions. And as you can see here on the graph, actually there has been some success stories as well. They conclude that it's time to increase the effort to save more species, but of course it's also nice to see that sometimes the targeted actions we do can help. So with that, we'll end my presentation.
Thank you. Next up is Anders Finstad. Please do feel free to ask your questions to Erlend on Slack. I guarantee he will be on there and answering all of them.
Okay, thank you. So I am Anders Vista. I am currently serving as a co-PI for the Living Norway Ecological Data Network. I'm affiliated with the Norwegian University of Science and Technology, and I'm a professor with the Department of Natural History, where I also serve as a data curator. And my particular responsibility at the moment for the Living Norway Ecological Data Network is in data standardization. Hence the title and the theme of this talk. And there's a recurring question that comes up when I talk about data standards and ecology, and that's sort of, this is not possible. And my aim with this talk was to show, yes, it is possible. Hence, the talk title should really be sort of, can we standardize ecological data? And the subtitle would be, yes, we can. But then again, that drifted me into the slogan, make ecology great again. And I sort of, no, that's not selling in 2020. Let's hope not. So anyway, this comes with a bit of warning. First of all, to the geeks out there, this will not be a very technical-oriented talk. It also comes with a promise to the non-geeks out there that this will not be a very technical-oriented talk. So, but first of all, needs sort of, what are we talking about here? So, with the talking about data standards, what really is this? I like to say that it's sort of three different parts. It's a content descriptor, which is what is this about, and we commonly refer to that as metadata. It's about the data structure, which is sort of the technical aspects, what's the file formats, and etc. And it's also about information content and retrieval of that, which is a common set of terms to describe your data, what you call your columns, and what do they contain, etc., etc. Now, that being described, as you mentioned in the previous talk, there's something that's called fair data. Well, it's a fundamental principle that in order for data to be useful for reuse, they should be findable, they should be accessible, and they should be interoperable with other data. As Alan pointed out in his previous talk, they need to flow together in order to do joint analysis, for example. And of course, they need to be reusable. There's several sort of things that needs to be in place to make this function. And it's important to note in this respect that FAIR is not a data standard per se. However, in order to make data fair, they requires a set of underlying standards in order to make data talk together, in order to make search engines find your metadata, etc., etc. So all of these points here need to have various different standards, and for ecology we operate with a different set of substandards again. So some standards are very common and some are narrowing down depending on which field you are working in. This is my take on what you say. This is the cornerstones of the data life cycle, which doesn't add up. But the data life cycle is sort of, we talk about data as going first being reusable, hence it's a cycle. Well, first it's the planning and collecting of it, which is the empirical part of it. Then it's the private things done by individual researchers, which is quality check, analysis, and storage. And then comes the interesting part, which is sort of new for the whole concept of open science and open data, which is you need to describe your data, you need to archive them, you need to share them with the world, and you also need to rediscover and reuse them. So it is often, the first part has been done by researchers repeatedly, but it's the last part here. We do the quality check, analysis, and storage, and then describe and share data. That's new to most of researchers in our field, and hence that's a lot of questions. What kind of standards should we use there? How should I do this? And it's also coming up when reusing data. How should I reuse data in a sensible way and an open, reproducible way? This is sort of what we're gonna try to tackle for ecology in the Living Norway network. Much of these issues are related to the perception of ecological data as very heterogeneous. I mean, it's a range of sampling techniques, a range of methods, and how could you possibly make these flow together? It's a problem that's been noted in the literature also previously, as this is a paper from 2019. And my claim here is that ecological data is not really different or disparate. They are just different traditions for how you express your data from different subfields of ecology. Either if it's coding your individuals in columns or in rows, or if it's raster data, or if you have them in a very highly normalized database, etc., etc. My claim to this is that there actually are common denominators for ecological data that make this very possible of sewing them together in a standardized way and sharing them together. So this I call a conceptual schema, or it refers back to what you call a data schema, but it's really a conceptual data model for how ecological data are built together. And I mean, everything we do for field ecological data and now sort of sample out in nature has a location. It might be a lake or an area, it might be a vegetation plot or whatever, but it has a location that can be georeferenced. At that place, there is something happening. An event, a sampling event, where someone goes out and observes something. It might be with a big research vessel, here illustrated by NTNU research vessel Gunneris, or it might be a very small research vessel, here by the NINA research vessel, MS Edna. Anyhow, it might be by a camera trap or automatic or remotely by radio telemetry. Doesn't matter, you do something there. Then you do an observation, which you term an occurrence. And it might be a batch of organisms, it might be a single organism, it might be evidence of an organism being there from a DNA or other trace, or it might be a picture from an automatic collection, or it might be some repeated measures of a radio-tagged individual. And you might do a sample of that again and bring back either whole specimens, part of a specimens, or specimens altogether. The point is that this is also different locations can be nested within each other. I mean, and events can be nested into each other, like, for example, the repeated samplings of the same plot. And then occurrences might have, there might be different scale you're observing on. It might be just nominal presence-only data, it might be binominal data, it might be an ordinary scale or true quantitative data, depending on. And at each of these points, you might have some additional measurements or facts of observations, either if the location has some names or whatever, or something about the event or of the occurrences. So this common set of describing data leads to the current practice where there are a number of ecological data sets shared today through data infrastructures, making them available and discoverable in a fair manner. And in other words, it is possible to do it. One of the reasons this is possible is that you have some data standards to build on. And this is where I get slightly technical and mention the most important data standard we had for this purpose, which is Darwin Core. And Darwin Core is a body of standards, in a broad definition, is a body of standards that are used and to facilitate biodiversity data sharing. In a very narrow definition, it's a library of a common set of terms that can be used to describe biodiversity data. It's maintained by the Biodiversity Information Standard Working Group, which is an international organization, and which is shortened to TDWG, or shortened TADWG. In the histories behind this is that this is the earlier taxonomic something working group. Now, how does this look? Simply a common number of terms. How would I describe an occurrence, for example? Well, you have a set of terms that refers to giving the observation and unique ID and a name of the organism, and you might have some measurements of quantity, for example, biomass, and it's etc., etc. Fairly simple example, but building up on this common body of standards, you're able to actually publish and disseminate quite complex data types. So, that being said, I am going to go through with you a few examples of what kind of data that actually are shared through the Global Biodiversity Information Facility today. Now, Dag Andresen going to come up next and talk more about GBIF, so I'm not going to give a brief more introduction to that, but just showing off that we have species inventory type of data, here exemplified by bumblebees and butterflies in Norway dataset from Nina. You might have panel data. Panel data is what we talk about when we say that this is repeated measures of a given individual throughout time, typically telemetry type of data. And you might have vegetation plots, like squares with a semi-quantitative abundance range. Or you might have line transects, either of the aquatic or terrestrial types. And of course, you might also have the traditional quantitative monitoring of animals or plants. Now, you might have less traditional methods, like eDNA-based inventories. And you might have camera traps, automatic detections of animals. Now, the main issues with this, or challenges, is yes, it is possible. We can share this type of data through Darwin Core as a standard, through the Darwin Core archive as a technical standard, and through ecological metadata language as a metadata standard for describing the data sets. However, a lot of these data are use Darwin Core in a slightly different way. So there's a heterogeneity in the application of how Darwin Core are used to code the ecological data. Just a brief example of 4 different randomly selected inventory type of data sets from GBIF, which all uses different types, uses Darwin Core in a slightly different way, meaning that they are not directly interoperable. The solution to this is, as we see it, that we need more community-derived recommendations, and possibly also more than recommendations from some of the funding agencies in order to how to publish and disseminate ecological data. This is an example that we've been working now through GBIF on how to publish sequence-derived data, meaning there's another word for eDNA-based data, and how to do that in a common standard format. Now, however, one of our main issues is, of course, still data stuck in the drawer. The lack of time, of resources, or of motivation or know-how of the individual researcher or organization to actually disseminate your data in a fair way is maybe the most significant hurdle we have. So our take on this is, how do we move forward? 3 short bullet points that we would like to address. We would like to build on existing standards which is Darwin Core, and the ecological metadata language as a metadata descriptor, and develop and employ community-driven best practices for verification of publishing and dissemination of ecological data. Second to that is to build on the existing infrastructure, like particular GBIF, to facilitate and develop user-friendly tools for easing the technical barrier for standardization of data and also reuse of data. And I guess Matt is going to touch on this on his talk coming up later in the session. And also, last but not least important, is to increase the community know-how of sharing and publish and how to reproducible and transparently reuse ecological data. That needs to target both existing researchers, both our new upcoming researchers and students, and also to a certain degree our funding agencies and managers. And with that, thank you.
So we do have 3 minutes for questions,
Yes.
if you're willing to take any.
Do we only have one mic?
No, there is one there that one of you can hold.
That's better. Like this? Yeah.
Yep.
Okay.
So there's a question from Chloe. To what degree do you think that we might profit or not from also standardizing data collection protocols, methods, definitions, etc.?
Absolutely. We could profit quite a lot for that to some degree, where it comes to some type of typical management driven sampling protocols for water resource management, etc., etc. It's already standardized. I think it's overly optimistic to believe that researchers or the basic research community is going to really standardize their sampling protocols, because this is going into details of their experimental analysis, etc., etc. Don't think it necessarily hinders reuse and interoperability and integration of data that the sampling protocols slightly differ, as long as it's transparently coded and disseminated in the data. Yeah, hope that was sort of an answer.
Yep. I think we have time for one more, maybe. Yeah.
Yep.
Question from Rainer. How does, for example, indigenous and local knowledge fit into these schemes and in your approach? This kind of data is crucial for moving forward in biodiversity related issues, and the knowledge of local and indigenous people must not be ignored.
Well, I guess it falls into the scheme as far as it's observed. I mean, depending on what's the observation unit here, that's kind of different. It might be the people's perception of nature you observe and code as observing, and their territory as your location, or it might be their observations that coded directly. That's kind of, I have no opinion on that particular, but in general, I think it fits, yes.
Thank you. Next up is Dag Andresen from GBIF.
So before I get started, I can also make some advertisement for the GBIF Open Biodiversity Data Ambassadors, which we have from Norway on the first row here, by coincidence. So you can all sign up to this, and you will find the slides. Are they auto-progressing? That I coordinated these slides very much with the GBIF scientific officer in Copenhagen, Dimitri Schigel, and you can do the same if you want to represent GBIF and do present GBIF in conferences like these ones. So I open with this one. So I'm trying now to describe a bit the context of GBIF in this open science, which is the topic of the conference, which is not that hard because that's sort of the main core mandate of GBIF, to provide the open science into the biodiversity data realm. In this aspect, we are trying to pull also the ecology data into this open science methods and solutions that GBIF provide. And I'm trying to show here that this is a wave that's coming, and that we have to start to address this. There is a huge shift in a paradigm shift in science, and also which is going to be reflected in the metrics that measure the result we produce and funding ultimately. This is moving fast. You have to grab on to the fire here to keep on. And we do need to develop different practices than we do today to be able to fit in and remain relevant in this new wave. And also, there is an expectation from society and wide to be able to access the data we produce and services that we maintain in this big data platform. And even more than that, before I actually go in now to describe GBIF, there is a shift in which GBIF is trying to contribute to. We were part of starting this data paper thing, where you can have peer review of data through a data paper, but we also are trying to influence the shift towards that. You can cite and have measurement of citation of individual data points and data itself without making it like into this data paper in between. So to introduce like a new currency in measuring science, citation of data, not only the citation of papers that describe the data and scientific results. And there is something you should know about. There's a Force 11, a task group that works on the principles to cite data. So that's something everybody should start to get into, and I hope that you will take part in driving this new way of doing science. So in principle, data should be cited, and if I manage to run through a long slide deck, towards the end I will show you a little bit our take, how we are trying to make data citable in GBIF and how we count the metrics around that. So, what is GBIF? GBIF is an international network. It's intergovernmental, so countries are signing up, and we aim to be an engine in this open science, to provide, so the main objective, the main rationale for creating GBIF is this free and open access to biodiversity data anywhere, online, for free, for anybody. There is a memorandum of understanding, which is not committing countries to do so, but we agree upon operating this together. And it's a distributed network, so each country must deliver a part of it. And we are the GBIF node for Norway. And we are now building a bigger consortium around this to include NINA and Artsdatabanken and so on in this deliverance of GBIF in Norway. Yeah, and we have the secretariat in Copenhagen. So that's a core team of people that service the whole network and work for us. Membership is increasing. So we just recently got a few members in the Caucasus and also now last week or something in Uzbekistan. But we are working to increase this membership. Each green country pays the membership fee. The blue countries, they are in the transition phase into the network. But, well, we hope to grow this, to color more of the landscape here. The primary object, Anders showed a lot about the different data types that flow through this network. But the primary point set here is the occurrence of a species in a location. And there are different ways that this is not into this common standard. You can have specimens, which is so I will show a little bit more also the individual of these data types, what characteristics for them. But we try to describe this occurrence of the species from specimens, from observations, from literature, where you can find evidence of the occurrences. You have the remote sensing, which Alan also showed a bit more on how you do remote this monitoring and these things. And Anders also touched a bit upon the eDNA monitoring. So I will show a little bit more about what is different from these things. But the thing is that we publish and show through the infrastructure the occurrence of the species. So it's a little bit simplified, and we need, as Anders was into, to look at how we can capture more richness of this, and that's what we're working on. But this common point of the occurrence of the species is used for a lot of purposes. So it's going out and filling in other information systems. So we are working now on a workflow, which is not operative, but we hope to get it into the EU directive reporting workflow to feed these reports through the data published in the network. The red list is, of course, generated off this workflow by Artsdatabanken. And there's work now also to join this more with EUCN and have more international data flows around this. There is a mechanism for the treaties and conventions where the volume of GBIF data is the metric for fulfillment of these agreements. And also we are working now on feeding also data streams and aligning them with research data portals, particularly the molecular sequence data. So the size of this now, we tipped up to 1.6 billion data records. There is a growing number now of data sets continuously growing. There's more countries joining. This is ticking up, and we also have new. So these are also a possibility for international organizations to join. So should be across more than one country. We have an enormous load of data downloads and access to the data portal. There is ticking up the number of data publishers. And also we still see a quite rapid growth in the number of cited papers that cites use of the data infrastructure. If you look at Norway, it's still also quite impressive numbers. 40 million data records, now 287 data sets, increased a little bit even last week. You can see the coverage of the data published out of Norway. It's not only Norway, but also data from other places. Yeah, and also we are not too bad on the data citation either. I will show a bit more breakout of that number. So it's quite scary to look at this enormous growth you can see over there. And then I put the autogram. So you can see that the proportion of specimens, so we started off primarily by being a service for the museum specimens, and with this enormous increase in other data types, we expect this eDNA to increase a lot. This ecological data could start really to influence the data volumes. But this means also that the museum specimens that was sort of the starting point is becoming a lower proportion of the data we serve. When you look at the taxonomy of the data, we share a lot of it. Here is birds, animals, plants, and other things. So with the eDNA, I will show this progress in a short while. We hope really to make this more evenly distributed across data types. And if you look at the type of data sets that you can publish on the network, you have metadata only. That's without data. You could pre-publish your data set before you collect the data even, and publish the metadata on it in GBIF. And then fill in the data later when you have collected it. Or it could be data which is not yet digitized, so only in paper, or even worse, on labels, which could show this data to the network and stimulate digitization funding and efforts. You have the species checklist, a list of taxon names, marine animals in Norway, things like that can help. Also, we have, of course, Artsnavnebasen, the complete checklist of known species in Norway, is also published in this data type, or data set type. And then the main sort of data type in GBIF is this species occurrence. So species at a place at a time. And then this, which becomes more relevant now for this ecological data type, is the sampling event, that you revolve the data publication around the events that Anders described. So I will try and show a little bit more into these different data. And before I go into these different data types, I will show that also a lot of the records now in GBIF is increasingly also this multimedia. So we have 66 million which have photos. It could be photos of the specimens in the museum, herbarium sheets and things like that. And you can see that from Norway we have also quite a good number there at 1.7 million. So it's also increasing up. There's also sound files and video files, and also some from Norway coming in. And I split it also up there by specimen, museum specimens, human observations, and these material samples. So if you look at what does it mean, these different data types, this basis of record, this type of evidence behind the species occurrence, the biggest piece of data in GBIF now is this citizen science observations. You have huge data sets like eBird with bird observations, pulling the numbers very high. iNaturalist is another very big data source. And from Norway, the absolute biggest data source is this Artsobservasjoner, the Species Observation Service. So these are enormous contributions to the growth in records. And the beginning, the origin of GBIF was more around these museum specimens, and they are getting richer now with photographs of the specimens, also to some degree now starting to be sequenced, so you can get access to the barcodes and also gradually other sequence data. And then you have also a very interesting monitoring of the scientific literature, where we try to where especially Plassey in Switzerland is extracting occurrences from literature, and also brand new species taxonomic descriptions that we can import into the GBIF very early on, straight off the scientific publications. So I have 5 minutes left, I think. One big challenge in this, as I mentioned early on, is also that there is a huge overrepresentation of birds. And then you can see this, if you try to look at the number of records against the number of species and so on, you see that when you go down the list there, insects is vastly underrepresented. So this is a challenge. I also tried to show this small icons there, that we have enormous 1.2 billion animal records, 300 million plants, 20 million fungi, 16 million bacteria and microorganisms in general, and only 0.04 million viruses. So that is something we need to address. And one way to address this is to start to look at, well, at this eDNA, this sequence-based data that Anders mentioned. So when you look at this biological code, this is a challenge. A lot of them, they don't have names. And to remedy that, we have started now to include the species hypothesis based on sequence data. So from UNITE, community around fungal data, they started to register species hypothesis which is registered with a DOI, and you could now start to mobilize and publish data in GBIF, which is tied to these species concepts. And also here from the Barcode of Life, we are integrating into the index, into the so-called taxon backbone, the taxa that you can report species occurrences on, these bins from the eyeball then, and so on. And Anders showed some of these eDNA data streams coming in. Magnify is one huge data stream that is publishing data on these environmental samples. And of course, Anders also took part in writing a wonderful guideline for how to actually publish this data into the network. And I think I can also show quickly the policy links. So the number of records in GBIF is part of the metrics that measure the fulfillment of countries of these treaties and conventions. So the CBD, the sustainable goals, IPPES and so on is based on and measuring the data published by a country in GBIF as one of the indicators for fulfillment. You have the Aichi targets. So GBIF contributes to a large number of them, and we highlighted here 4 of them. Should I take the last, or should we take 1 minute? Yeah. So we contribute to sustainable development. You can see here the growth in data publication, and Norway is contributing some to that list. We would like to see that number increase a bit to be in proportion to the data we produce. And just so that you should all start doing this, it's very easy. You download data from GBIF, and together with the data download, you get a DOI, which you cite in your work. So that should get you all started doing this. So I don't know, do you have a question?
You don't have time, but you can answer them on Slack.
Yes, I will. You should say.
Just a quick housekeeping note. The questions on Slack, they will be answered. Maybe not all right now because we're also listening to the talks and everything. So, but they will be answered. Next up, Matt.
Okay, it's quite a nice smell, so I might be high by the end of this. That's of course not to go. That's why. There we go. Okay, so I'm Matt Granger, postdoctoral fellow here at NINA, and I'm going to talk about Living Norway R. So Living Norway R is our companion package for the Living Norway project. It's still in development, but we want to introduce you to it here and maybe get some feedback on what we can do and what you want. As ecologists, we tend to have a workflow that's represented by this schematic. So me, I'm interested in pheasants and peafowl. So I go with my binoculars, I go watch some peafowl. I come back to my office and I start to put in my data. Within my computer, I might do my modeling and then do some graphics. Write that up as a paper, and then I push that out over to here to be published somewhere, hopefully. That journal I work for might want me to, the journal I submit to might want me to share my data. So I could go back to my computer, spend hours working out which version I used and all this sort of things, reformat my data, put it on some sort of server, Dryad, Figshare, whatever. More often than not, some of us might even just put this little statement in the end of our paper saying data available on reasonable request, and hope and pray that no one ever asks you for that data. But there's people around the world who do ask for data. They're after open science, open data. And this tweet here is someone talking about how that interaction goes. Them, you ask for some data, there's a reasonable request. Sorry, I can't give you that data. So this happens quite a lot. I'm very interested in code and code sharing. So earlier this year, there was a paper in Nature, a very interesting paper. I wanted to find the code so I could recreate their analysis. When I looked at the data availability and the code availability statements at the bottom here, what they say is that there is code available, but that code is actually the packages that I just make the things. It's not actually their code. It's just the package they used, which is not very useful for recreating someone's data. It's not reproducible. Erlen mentioned a few papers that have come out recently. This one's very recent, and you can see that actually we are sharing data. So we're doing well. Of 346 papers that looked at, 80% of them, roughly, had shared data. When you look at the code, it's less good, but it is improving slightly. So about 30 or 40% of the code is being shared. However, all the data might not be complete. So from another publication from 2015, they looked at the top-ranked journals in ecology and evolution, and they looked at what data was being shared. So they looked at the papers, and they found that only half of the papers they looked at shared all of the data from the analysis that they'd done in the paper. So this plot here just is on a scale of 1 to 5, so 5 being very good, everything's shared, everything's there, 1 being rubbish. Even when the data is shared, sometimes you can't reuse it. And there's lots of reasons for this. It could be that it's using, it's just a photograph, an image of the data. That's something that often happens. So you can't reuse the data. You can't make use of it. So even though it's being shared, it's fundamentally useless. So I just have to change over. So this is results of a survey from Wiley in 2014. And it asked the question, what are the barriers to sharing data to different researchers across the world? And I've put them into these boxes, or circles, of different sizes. The size represents the number of people who responded. And I've put them into different categories. One's in society, so things that we as a society have to deal with. I can zoom in there. So it's things like concerns about confidentiality, concerns about ethics, perhaps. People worry about being scooped, so having their data used and their famous paper being taken away from them because someone else does it quickly. Misuse is obviously something that people worry about. Lack of funds, and it's not required. There's a couple of things I put into this researcher box. So these are things that the researchers might have to deal with. Data's not interesting. So if your data's not interesting to you, then why would it be to me? But we can still share it. And not my job. I think that's an interesting one because a lot of our data we work with, is publicly funded. And I think it probably is our job to be sharing this data. It's not ours to keep all the time. There's a couple of these things, though, quite a big proportion that Living Norway and Living Norway R can help deal with. So here, we can help to deal with where to share your data. We've spoken about GBIF this morning, that's one option. How to share your data, so that's something I'll talk about now. Having no time, so finding ways to actually integrate sharing data within our workflows. Okay, so it's not an extra task we have to do. And improper credit. Doug just talked about the citation process for GBIF. So that's something we can use to improve our data sharing and help us get benefit from it. This is the future. This is the future we envisage for Living Norway and Living Norway R. So this is our workflow. So again, I've gone out and surveyed my peafowl or my pheasants. I've come back to my office, to my computer, and then I've looked at, I've entered the data. But this time what I do is I curate, in that orange box, an archive. Now that archive can sit on your computer, it doesn't have to be shared, but eventually it will be shared if you want. But that archive, what we can do is then use that, a fixed, non-changeable data set that you can call back into your working environment and do your modeling and your analysis, and also some aspects of writing up your data as well, which I'll show you in a minute. And then we can send that all off to a publisher. So here we go for the Living Norway R package. The idea is it's an R package designed to facilitate that workflow and help people, make things easier. The main features we're going to have, or have already, importing and exporting Darwin Core archive files, mapping data to the Darwin Core terms. There's some R packages that allow you to map your data to Darwin Core terms, but they're quite difficult to get used to, almost impossible sometimes. We want to make that process a lot easier, smoother. Automatic metadata extraction and writing functions. We're going to have those, so all the metadata aspects that you want, we can just pull straight from your data set. Some plotting functions, but also some modeling functions. So to help people integrate this whole thing into their workflow. So I'm going to give you a quick example of some of this, and there is R code, I'm sorry. But we're going to talk about one of Erland's data sets on rock ptarmigan. So we've downloaded this data set from GBIF. It's publicly available, and there's the DOI to it. It comes from a publication, the paper's linked there. We also have some of the code for recreating the analysis in that publication is available on the Open Science Framework, OSF. So I used this data, I downloaded it a couple of days ago, and ran it through the workflow in Living Norway. The first step in Living Norway is to set up your data package. So this is a set of files where your project you're working your project from. And in that data package, you've got several different folders, let's say. The first one is going to be your data. So you've got a place to put your raw data. Any scans, any extra data you might have, so things that come from the field. So if you've handwritten field notes, you could scan those and put them up. But also some derived data, so anything that you have to do some coding to adjust your data, log transform, whatever it might be, some extra things go on there. You can save it all in there. Got a data management plan, this DMP in there. Let's move that down. Some metadata. And the metadata is important because that's in 3 formats. One is the XML metadata, which is computer readable. There'll be an RMD, so a markdown file, which you can also read as a human. And then there's also this file which actually tells GBIF which files relate to each other in the end. Okay. And then you can also put in your scripts, your analysis scripts, in the folder somewhere. Okay, so that's the structure of the folder. We're using object-oriented programming, so it's a new one for me. So Joe's been doing a lot of this work here. What it allows us to do is create an object that is in R, that is a representation of one of these Darwin core main terms, these things. So we have this, for example, an occurrence data set, and we can map our occurrences from our raw data directly onto the Darwin core terms that are used. And if, like me, you're not used to the Darwin core terms, Joe's written some brilliant code to get us actually almost like a glossary of terms. So if you don't know what the term means, you can put in some code and it'll output what the term means. So here, an occurrence is an existence of an organism at a particular place or time. Okay. So for each different type of term, you can search the terms and get an output of examples, where you can find where that term was defined, and how it's defined, and any updates that happen to it. With these objects, you can actually make use of the data in the same way you would do as you would do normally using a data frame. So you can actually export a part of this object, the raw data that you want to use, or the data you want to use, as an object. Okay? So for example, if you want to do simple things like summarizing the data, so here I've just got this rock ptarmigan data set, and I'm summarizing the sex. So up here, I'm making it exporting a data frame from the object, and then I can use that to do other things. Also, with these object-oriented, we can actually embed functions within our objects. And that allows us to then, it's neater and easier to have that. So we can, for example, if you want to get the bounding box, so the geographical extent of the data set, you can just type in rock with a $ get bounding box, and it outputs the bounding box for you. Okay? So that will go into your metadata. Making maps is made quite easy. You just use the function plot map, and it gives you, in this case, the leaflet map, but we can change that to a ggplot or whatever map you would like. So by using the object, it just calls up the underlying data and plots out the map for you. There's a few extra things we can add to this, but it's really what people would need. So we need to know what you want to add to. We're quite early in the stage of development here with the metadata, but we're starting to build an object, which is a metadata object. We can use that to either call in from a already published Darwin Core archive on GBIF, or we can create our own. What it allows you to do is to get the information, that metadata information, from the Darwin Core archive, and you can use that in some way. So here, it's not very neat at the moment, but we call out for the abstract, we get the abstract of the data set in. And you can use that in your RMD file, so your markdown file, or in your report, whatever you want to do. Then you can do some analysis. So all within the same object, you can do some analysis. Okay? So here, because of occurrence data, it's often things like survival we might be interested in. We've got this function for running a survival model. This is still very much in development, but we can run a survival model and output to you the survival. And we can add a plot to that as well, if you want to. So this is one of the things I'm quite excited by. So we can start to have ways to help you write up standardized results. This is particularly helpful for things like working with monitoring reports, annual reports, where you have the same basic structure. We can embed our code into Markdown files that allows us to pull out those key bits. So we can have templates that will allow you to write up your data for those structured areas, particularly for certain analyses. This just shows what it looks like to the human at the top. As you're all humans, you'll see that. And then below there, there's the results under the covers, which is the R code that goes into that. So finally, we're still in development, and we really want you to tell us what you need and what you'd like from us. So there's 3 ways to do this. The first is put up an issue on GitHub and tell us what sort of features you'd like to have. The second is to contact us over the next couple of days on Slack. So it's either Joe, myself, or Erland you can contact, and we'll try and add those issues later on. Or you can just email me, old-fashioned. Pigeon post might work too. So thank you very much, and hope you enjoy the rest of the colloquium.
In the meantime, we do have a question. We have time for a question, so let's see, there came in a question just now. Are there plans to support Darwin Core extensions for sequencing data like Mixes extensions in Living Norway are in the future?
Yes, there's lots of plans for the extensions, and it's something we're looking at. At the moment, it's just in development, and we can let you know how that goes.
And then there is another one. Our code is very useful as the underlying code, but in most cases it does not work to ask researchers to enter metadata in an R script. Do you have a front end to enter, edit, check the metadata and its consistency with the data?
Soon. That's one of the things I'm working on now. We're trying to get that to work, to make it a lot easier for that process. If anyone has data they would like me to test for them, that would be wonderful as well.
Thank you. Next up is Caitlin. I'll do that.
Everyone's used to it now. Yeah. Great. Thanks. Okay. Hello. My name is Caitlin Mandeville. I'm a PhD candidate at NTNU here. Working with Anders and Ireland, who you've heard from already. So yeah, I guess I'm closing out this first session, talking about some of the same themes as the other speakers have so far, but kind of turning and trying to take the perspective of data users, researchers who are working with different kinds of data. Let's see if I can get rid of that. Yeah, so we're going to do this by sort of taking the perspective of one specific data type, presence-only data, and using that as a case study to look at some different trends in reproducibility and openness of studies that use biodiversity data as a whole. And I should say most of what I'm going to be presenting today comes from a paper that's in review right now. And the three co-authors I have on that paper have already talked to you today. That's Wouter Koch and Anders Winstad and Arlen Nielsen. Yeah, so what we did here, this work kind of arose from a systematic review where we tried to comprehensively look at any and all peer-reviewed English language articles that were sort of connected by one common thread, this use of presence-only biodiversity data. And by that, I mean point occurrences of a species or organism observation that have just these two key elements, the taxon identification and the location, no absence data. So our interest in this topic actually didn't really initially arise from looking at open data as much as it did from this top left element, citizen science. So most of my research right now has to do with opportunistic citizen science data. And as you've heard already, that's one of the major sources of presence-only data and open data, along with historical data that's being digitized. So commonly, these data sources in recent years have been digitized and uploaded and shared in these open online data platforms, with GBIF being a major example of that. And that has sort of facilitated this massive growth in the analysis of presence-only data. Of course, that's not the only data type shared in platforms like these, but it is kind of a major one and has been increasing in analysis lately. So we started sort of looking at this question from that angle, and we began to think, are there other sources of presence-only data that aren't being talked about as much? Are they also being shared openly, or where are they applied? That kind of led us into this question. Presence-only data is also interesting because it comes with all these challenges for analysis. And there's a lot of ongoing innovation right now in the best approaches for working with this data type, including a lot of research by colleagues here at NTNU and elsewhere in Norway. So we were sort of interested in seeing what analysis approaches researchers on the ground are actually using. And then finally, and what I'm going to mostly focus on today, presence-only data is pretty interesting also from this open science perspective. So it's probably one of the most straightforward types of data to share with existing infrastructures and best practices, and maybe the most talked about in terms of sharing. So we were interested in sort of using it to test how consistently some of these open data practices are being applied in the literature as a whole. Yeah, so really briefly, we retrieved just under 3,700 articles from the Web of Science. And we searched for them, targeting all abstracts whose text contained both one of about 30 terms related to the presence-only nature of data, and one of 5 terms related to biodiversity. So we retrieved these articles, we screened their abstracts, and we retained just under 2,000 articles. Then on that full data set, we did some bibliometric analyses and some topic modeling to kind of get a sense of broad trends in the literature. And then we also took this subset of 300 articles, which we then read in full and collected some characteristics, especially some characteristics sort of relating to their openness. So I'll present some of our results. Really broadly, we saw, as expected, that the application of presence-only data is increasing really dramatically, especially over the past 15 or so years. And by breaking the full data set down into these topic categories, we could see that the nature of publications using this data type have kind of been shifting. Initially, the growth was primarily led by this blue color methodological papers. And then you can see right around 2015, the increase in method papers sort of flattened off, and the growth has really been driven since then by applied papers and data papers. And we see similar trends in citations, with that shift happening sort of right around the same time. We're kind of interested to see and maybe speculate with this lower relative citation rate of methodological papers more recently, whether it's possible that might imply that applied researchers are sort of still more often citing older methods papers, and maybe newer methods aren't catching on quite as quickly, or whether there's some other trends going on there. But then broadly speaking, who are these academic users, these researchers writing papers using presence-only data? Well, one of the first things we saw that it's a really large and decentralized group of researchers. In the 2000 papers that we looked at, there were well over 7,000 distinct authors in nearly 500 distinct journals. So very spread out. And the average author contribution was relatively low, so we're guessing that these are mostly authors who don't only work with presence-only data, but who just sometimes incorporate it into their broader research. Just a small number of highly cited articles. And the distribution is global but unevenly distributed, which sort of maps up with the figures that we saw in previous talks today. That distribution is really centered in Europe and North America right now. Yeah, so this is kind of the big picture that we came away with of what this pool of authors working with this type of data looks like. Broad, decentralized, and growing quickly. So then to get sort of a closer look at the practices that these researchers are actually sort of engaging with, working with this data in the literature, we turn to that 300 paper subset that we actually read in full and collected data from. So one of the first things we looked at, showing the results of here, is where researchers were accessing their presence-only data from. And right off the bat, we sort of expected that a lot of this would be from open databases. It turns out that that is the most common source of presence-only data, but it's far from the majority. What you see in this histogram here is, of course, the various sources that data was accessed from by the papers in our review. And I should point out that it's not mutually exclusive. Some papers access data from multiple sources, and that's reflected here. So yeah, you can see that open databases represent about 43% of the papers. But there were a lot of other sources that papers accessed this presence-only data from. Next on the list, you see original data collected by the paper authors, the literature, museums, and so on. So then the next question that we kind of asked ourselves was, so researchers who are publishing these data from other sources, are they then making those data open? Is this generating new open presence data that other researchers can use? And by and large, we found that this was not the case. We are looking here at all of the sources of data that we just looked at on the previous slide. And for each article that accessed some data from one of these sources, what percentage of those articles took different publishing practices with the data? The dark blue color represents data that was published using fair data principles. As you can see, that's really in the minority. The medium shade represents data that was published in a more limited way, like, for example, in the appendix of a paper. And then the light shade is data that weren't published at all. So there's a lot of work to do in this area. It's interesting to me that these numbers are quite a bit lower than what we just saw from some previous studies. Maybe that has to do with the journals that were included, a really wide range of journals, rather than focusing on a few more well-known journals. But an interesting trend. So then one of the next things we considered was sort of the metadata that the papers described of the data sets they were working with. And in this kind of topic, we really focused in on the structure or original sampling design of the data. It's an interesting question because when papers describe to a greater extent the sampling design, it can open up more doors for different analysis approaches. It's often thought the presence-only data sort of go hand in hand with being opportunistically collected, but it's not always the case. That can be collected in a structured way, and we wondered how common that was. So, as you can see, opportunistic data is of papers that do report that sampling design is the most common, but there's also quite a bit of semi-structured and even quite heavily structured data that's being analyzed in a presence-only framework. And perhaps not surprisingly, there's also quite a bit of papers that did not describe that data structure at all. And that distribution of reporting structure was pretty evenly distributed throughout those different original data sources. Interestingly, and just kind of as a side note, we were interested to see that even pretty simple elements of the presence-only data were not always reported by the articles, like in the example of sample size right here. Yeah, so getting back to a discussion of structure, we didn't see as much of a difference in analysis approach the articles taken, driven by the different data structures as we might have expected. More structured data does correlate with more inferential analysis approaches as opposed to summary statistics or sort of just reports of data, but it's not such an extreme correlation. And opportunistic data actually has a pretty high proportion of articles that were engaged in some sort of rarer inferential analysis approaches, possibly because there's so much innovation going on right now focused on opportunistic data. And this is just kind of an overview of overall the different types of analysis approaches that we saw in this data set. You can see that species distribution modeling is definitely dominant, but there's really a wide range of approaches that were taken. So yeah, that's kind of the landscape, the big picture idea of what we saw when we were looking at this presence-only data set. We really were sort of surprised by some of the findings with regards to the openness of the data that we were looking at. One of the major conclusions that we came away with was that there's not such a tight relationship between presence-only data and open data as we might have expected. And we wanted to kind of dig in from a user perspective to see why that might be, and why some of these data maybe aren't being shared as much as we thought they might be. So to do that, we subset our data into these 3 different categories to kind of look at a profile of 3 different users of presence-only data, defined by where they accessed that data from. Users who access data from open sources, those who only use their original data, and those who used what I'll call found data, which is data that they didn't collect themselves or get from an open source, but maybe from a management agency or a nonprofit or the literature. So I'll just walk through some key facts about each of these different profiles really quickly, and then sort of try to draw some overall conclusions. Looking first at users who collected data from an open online platform, that's 43% of all the papers we looked at. We see that just over half combine their open data with data from other sources. So I'll have a little ticker going here, kind of compiling the information that we're picking up. That's the first piece of information. We were surprised next to see the great variety of different open data sources that these open data users accessed. In the about 130 articles we reviewed that used some open data, they accessed it from 120 different sources. So very diverse. We called 8 of these large databases, basically meaning that they covered data from a very wide spatial extent or very big taxonomic scope, or both. And 57% of open data users accessed a large database. The rest we considered small, and actually a slightly greater number of open data users actually accessed a small database. Of course, some accessed both. We were interested in this fact because there's research that shows that small databases might not remain as stable into the future, or might not keep up with best practices for data standardization. So it's interesting to us to see what a big role they play in users' access of open data. Most open data users, just over 2/3, use just one open data source per paper. And in general, these studies that accessed open presence-only data, about 60% were small or regional, while the rest were large or global. Looking really briefly at this subset that exclusively used open data, we see that just under half didn't report any structure on the data. And in some cases, that may be because they didn't have or didn't know how to access that information. The bulk of the rest described the data as opportunistic. Turning then to look at this other subset, the 56% of open data users that integrated their open data with non-open data, we hypothesized that this set of users, because of their proven familiarity with open data, might be more likely to then themselves publish the data that they obtained from other sources. We found that that really wasn't the case, kind of surprisingly. Only 24% of these users made their non-open data available after publishing their analyses. So yeah, to quickly sum up our profile of sort of an average open data user of presence-only data, it's great to see that there are many users accessing this data. we were interested to notice that many of them were relying on these sort of smaller data aggregators, and they often don't share their own data, so definitely opportunities for continued discussion and education. Moving along to look quickly at the profile of a user who is sort of on the opposite end of the spectrum, collected their own presence-only data and analyzed it in that framework. A slightly smaller percentage of these made their data available, 19%. On a positive note, the vast majority explicitly described the structure of their data. And nearly all of them were small or regional in scale. On a potentially related note, these studies were almost 4 times as likely to contain themes that explicitly related to conservation interventions and decisions than applications of similar data from open sources. That kind of sums up the profile that we're looking at here. Thinking about reasons that might hold these users back from sharing their data, certainly many of the discussions that we've heard earlier today could apply here. It's quite possible, sort of thinking back to Anders' talk, that because some of these data do tend to be semi-structured, it may feel like more of a barrier to these researchers to share them. Or especially if the data have already served some clear and explicit purpose, specifically being tied to local conservation outcomes, the researchers just might not have the motivation or time to take it further and then share those data. It's hard to know, of course, exactly what the reason is from this data that we have here, but it's clear that this is a pool of data users that's producing quite a lot of data with the potential to be shared. And then finally, we'll quickly go through this profile of users who access what we're calling found data. In many ways, their profile is very similar to users that are collecting their own data. About 18% are sharing that data. Tends to be, for the most part, smaller regional in scale. And again, more likely to be related to sort of direct conservation outcomes. The major difference here is that fewer of these authors are explicitly describing the structure of their data. And given that this is data that they've accessed from other sources, it's very possible that in many cases this is because they just don't have that information. So we can speculate that possibly that lack of complete knowledge of the data's original structure or sampling design may in some cases hold them back from sharing this data, along with issues of possibly not knowing where they stand with data ownership, given that they haven't collected it themselves, and all the other issues that we've discussed already today. But again, a major source of data that these data users are going to quite a bit of effort to collect and compile from non-open sources, and would be very valuable, of course, to be openly shared. So, the main takeaways that we see here in terms of open data, the good news, of course, is that 43% of present-only data users are looking to open data. So that's a very positive thing. Not all of them are yet practicing best practices for data sharing, and many of them are relying on databases that may not be as reliable into the future. Additionally, a great deal of presence-only data are going unshared, so this represents a really great potential moving forward. In particular, studies with direct conservation applications or smaller studies tend to be negatively correlated with data sharing. So, on the flip side, this means that this is a great audience for continuing to normalize the sharing of ecological data. And we expect to see that as interest in presence-only data continues to increase, researchers are just going to keep using this data more and more, and this really represents a great opportunity for continuing to normalize data sharing among that audience and with this type of data. So that's all I've got. Thanks again to the co-authors and to you all for listening. Thanks.
Thank you very much. That concludes the first session. We have about 5 minutes to get ready for lunch. So I suggest we do that and be back here on time at 1. And don't forget the survey. we need those answers. Okay.
Welcome.
Morning or good afternoon.
Let me just. Okay, we're having some feedback here because we're two different levels also. There. Let's see how that works. Now, there, we're on. Hello, yes. We're sitting here in a small technical room and there are speakers and microphones everywhere, so we had to mute the others.
Okay, sounds good.
So good to see you. Welcome.
Yeah, you too. Thank you.
Are you all ready and set with your presentation?
Yes.
Yes.
Yeah.
I have on my schedule here that you're on at 1:20 Norwegian time.
Yes.
It's in 35 minutes or something.
Yep.
Yeah. And will it be only you or with John Feiberg?
John is going to, I think, log in.
Okay.
But then not. I think I'm doing all the speaking and I'm sharing my screen, sharing the slides.
Cool. Anyway, the sound is very good and the image is perfect.
Great.
So perhaps we should test sharing the presentation.
Sure.
Just to make sure.
Thank you.
If you're hearing some noise here, it's because the big hall is also connected to the same meeting here. So.
Okay.
Yeah.
So this is, I have to use this program, so it's a little different because my slides show on the left instead of being a full screen, but does that look okay otherwise?
Sure, it looks okay. But you want this thumbs on the left to show, or is it possible to perhaps?
It's not possible because when I export this to PDF, it's not working correctly.
Okay.
And if if I make these disappear, then I can't advance through the slides for some reason.
Okay.
So.
Sure. Right. Because otherwise, it's supposed to be possible to share an application directly.
Is that not what it's doing? That's what I did.
Yeah, because there are two different share options, and we have time to test them if you want to.
Yeah.
Okay. Yeah.
I did that. I did share application, and then I shared just my keynote slides.
Right. Okay.
So I don't.
Then you're probably right. Then it has to be like that.
Yeah,
I know with PowerPoint, it's two different possible options for sharing here. You could either share the entire application, or you can just share whatever is viewed on your screen, like what I'm seeing here. Yeah.
So I think it's because it's Keynote. Maybe it's. I tested with a WebEx yesterday, and this is the best I could do for sharing.
Fine.
So.
And it will be more than large enough, and it will be good. Perfect.
Okay, and this is as small as I can go with the making the slide small.
Okay, I understand. Okay, so good. Let me just see here. And welcome, John Feiberg.
Yeah.
Yes.
Thanks.
Hi. My name is Lasse. I'm supposed to be hosting this technical part of the WebEx meeting. Besides from that,
thank
I'm not
you.
really. So
Yes.
I hear you. I hear you very well, and I see you. And you will not be sharing anything.
No,
from the screen.
I'm basically here to support Althea and help answer questions and be involved to the extent that is is called for, I guess, for the day. But I'm excited to hear the talks in the session too.
Very well, very well. What we will be doing here is that you cannot share your presentation before the previous presenter has finished, because otherwise you will be stopping the other presentation and showing your own instead. But you will not be following the stream at all now, will you? You will only be following the WebEx meeting, right?
Mm-hmm.
Yeah. So then you will see when the previous presenter, Ingrid Hageland, is finishing up, and what we have been doing is giving a 15-20 minutes talk, and then given enough time, there will be a few questions after the talk. Don't share your application right away after we're clapping and the talk is finished, because the chair here might have a couple of questions for Ingrid before it's wrapped up. And we will be streaming pause image here while you're setting up. So we're calculating like 10, 15, 20, 30, whatever seconds between the two presentations, so that you can share your screen and everything.
Great.
Mm-hmm.
Sounds good.
Yeah, sounds good.
Okay, very well. The one before you will not be on this WebEx meeting, but will be here on site after. But the one after you, but the one after you will be on WebEx. So we're still waiting.
Yeah, it's unfortunate we weren't able to travel this year.
Yeah.
But
that's just how it is, isn't it?
it is, but it's nice to be able to participate, even so.
Yeah, we're very happy you could join us anyway in like this. It seems like we will have this situation for a while, so we just have to get used to holding conferences like this, I guess. Are you hearing quite a lot of noise on your side now?
Well, we could see I could see in the background people walking and coming back in.
Yep, because the room will start to fill up now in the next 10 minutes. So I guess in a few more minutes it will be. We will more or less hear the people small talking on their way.
Yeah.
Yeah.
Can I ask one question? I don't know how much I'll be able to tune in this afternoon, but if we wanted to tune in this afternoon or tomorrow, is the best way to do that via Slack?
Yes.
Okay.
I would recommend following the YouTube stream on Slack.
Okay.
Yeah. And also the direct web address to the YouTube stream was published this morning in the Slack channel.
In the Slack channel, okay.
Yeah. So you'll be able to follow it.
Okay.
Althea, I lost your image.
I know. I don't know why. Can you hear me still?
I can hear you.
I think I can still share my screen. I lost everyone's image for a second.
Okay, I got the error message due to low bandwidth, and then it disappeared.
Can you see my slides still?
I can see your slides.
Okay.
But there is an error message popping up once in a while here, and it seems like it has something to do with bandwidth on your side.
Yeah, I changed. Now I'm on my hotspotting on my phone. Is that better?
Now you're back again.
Okay. Sounds good.
Okay. Okay, but then let's hope it stays like that. And we got Rico and we got Image, and you're all ready to present.
Okay.
So
Thank you.
I will mute you now, as
Yep.
the others are joining in, and I will make sure to unmute. There you did it. Good. I will make sure to unmute you when we're starting. And do you want me to let you know when to start presenting, or are you just following this and you go whenever you see the numbers?
I guess so if the person before me is speaking in person, can I still see that through the WebEx meeting?
You can try. You can see it on the WebEx meeting where it says Trondheim Amphi.
Okay.
Yeah.
Okay. Oh, got it. Okay. So
Yeah.
that's
So whenever she's leaving this table, then it's all set to go.
okay.
I can anyway make sure to alert you.
Okay. Thank you.
Yeah. Okay. We're still waiting for the last person to join in on the meeting before we go mute on everybody.
Okay, that should be fine. Yeah, that should be fine.
Okay. I will mute myself for a short while too. Okay, so both Thea and John, I lost image for John, but perhaps that was on purpose. Sorry, you're muted. I unmuted you, John. Yes.
Yeah, I just stopped my video because I didn't know if I should. I saw the others were all in the background, so I don't know, whatever you prefer.
Yeah, the two of you are more than welcome to stop your video until you're presenting if you want to. That's fine. We're actually still waiting for the last person that was supposed to join this meeting to present after you.
Okay.
So we're just hoping that she is entering at a fortunate moment. But I will make sure to be alert here and mute her at least on the moment she enters the meeting. So, yeah, so we're just counting on this to be perfect, I guess. Anyway, thank you. I will stop my video as well and see you later.
Okay, welcome back. Hope you enjoyed your lunch. We are ready to go with session 2 of the Living Norway 2020. And first off, we have Ingrid Hegland from the NTNU Library. Looking forward to it. Thank you.
Thank you. So, first of all, thank you so much for inviting me. It's been a real treat to be here so far. So, I work at the University Library at NTNU, and you might wonder why I'm here. Well, one of the reasons is because at NTNU, it's a library that we're trying to promote open science. We're trying to support researchers doing open science, doing research management, and think about best practice in research. We have basic support, so I also represent the research data at NTNU team, and we're the library and IT together, trying to give basic support and infrastructure to researchers. But now I want to talk a little bit about what, why, and how open science. And we've already kind of discussed this earlier. So this is just taking a step back, saying, why should we care about open science, and what do people mean when they say open science? And I want to argue that as a researcher, you are already doing open science, at least to a certain degree. And I've visited most of the institutes at NTNU talking about open science. And to be honest, you guys, you are doing pretty much open science compared to a lot of other disciplines and research fields, just so you know. So just to start out, what about defining open science? So there's not one definition everyone agrees on. It kind of depends on context or who is talking about open science, but there are some things that are common for many of these definitions. One example, to make the outputs of research open. That's one definition. That may be a narrow definition. Others say that the principles of openness should be expanded to the whole research cycle. So everything you do as a researcher should be open if possible. I would say that the second definition is more close to what we work with at the library and at NTNU, and you guys as well, as I have seen. And also the Research Council. They talk about the processes and the results being open. They also emphasize promoting quality and knowledge development. It's not just about shoving a PDF out there, it's about the whole process, how you develop knowledge. And the umbrella term open science, it can be a lot of different things. So this is a suggested taxonomy for open science. I won't talk about all of these, but I will mention this one that we have seen earlier, which are the main core. That's the publications, maybe the most familiar one for many fields. We have talked a lot about open data today, but also elements like open source, sharing your code, your analysis, open notebooks, so sharing methods, work in your project as you go along, but also the open peer review. So how do you evaluate research? Should that also be open? Scientific social networks, the collaboration networks, how you actually work together, what tools do you use? And also open educational resources and citizen science, involving even more people in open science and the open science practice. See, these are some of the facets. If I were to define open science, I would maybe say it's open and free sharing of knowledge in a digital world. Because this is important. We have new tools, new possibilities. The way we do and the way we share science has changed in the last 100 years. And I also want to emphasize the parts responsible, reproducible, and accessible. It doesn't mean open just to be open. It means open to promote good science. It's always as open as possible and as closed as necessary. So at NTNU, we work with these 4 core values for open science. It's about collaboration, working together, building on other people's research and findings, dissemination, sharing what you did, how you did it, what are your conclusions, transparency, showing what you did and how, and reproducibility. So hopefully I'll use these terms are not really new. We have for many years talked about research ethics. It's an important part of research. And what I just mentioned related to open science, research. So I hope that we will stop talking about open science and it's just simply science. It's good science done in an open, transparent way. Sharing your methods, your processes, your results. And what I see here today, I see that we are really taking steps towards that. Although we still have a way to go, of course. So this is some of the ways we talk about open science and why open science. I mentioned the quality and integrity, making it more visible what you are actually doing, make it showing what you did, transparent research. But also from the funding side, it's also about making the most out of the money we spend on research. So economic benefits. If we can re-share data, reuse data, maybe you don't need to spend that much money on new projects. Open science might make research more efficient. If you get your output out there quicker, it can be reused quicker, it can be combined in different ways, it might help innovation, and we might have global benefits. So these are all these big, hairy goals for open science. And of course, public engagement and trust. Showing and being transparent what you do, how you do your research, it hopefully increases trust, both within research communities and towards the public. And one of the reasons we talk about open science or reproducible and transparent science is that many claim we have a reproducibility crisis. And I think this will also be addressed later today. But I just want to mention this study asking different researchers in different fields, do we have a reproducibility crisis? And over half of them said, yes, we do really have a crisis. They also asked researchers, so are you able to reproduce when you have tried to reproduce experiments? Could you reproduce? And over half of them said, no, not even my own experiments. I couldn't even reproduce my experiments in my lab. So we have a problem. Many reasons for this problem. Some state, for example, p-hacking, selective reporting, things like that. You want to have all these really cool, exciting results, so you might not publish the unexciting results, for example. But also sharing what you actually did, sharing your data, sharing your code, so others can go back and check it and look at it, reanalyze and reproduce. I think this is an important part of it. I just want to give an example of very non-producible research. You might have heard of it related to COVID-19. So there was a Lancet study looking at the effect of one of the medications, and then it was retracted. And one of the reasons it was retracted was people started digging into the actual data behind the publication. So in the publication, they said, oh, we have this huge data set with data from hospitals all over the world. And then they started asking different hospitals, different countries, so how did you contribute to this data set? And it turned out they could not get any confirmation at all that anyone contributed data to the data set. So suggesting that the data set didn't really exist at all. And then two of the co-authors said, well, we haven't seen the data ourselves, so I think we have to retract. Which is pretty amazing that they never saw the data. So then Lancet said, oh, we are learning from this. We have now new guidelines. So when researchers submit the paper to us, at least 2 of the researchers have had to look at the data set. Not very impressive, but at least it's a step in the right direction. So an example of a journal that is a bit more forward-thinking is PLOS ONE. If you submit the paper there, you have to tell where can you access the data if you want to look at it. There are, of course, exceptions related to privacy, but you cannot say just data available on request. That is not good enough. This is one example of working towards more transparent and more open science. And I want to mention the top guidelines, transparency and openness guidelines. So these are guidelines where you can go and check, what about the journal I want to submit to? What about my journal? How are they doing with data availability statements? What about pre-registration, code availability, and so on? And you can see how stringent are different guidelines for different journals. And this is really an important way for journals to help promote open science and transparency and reproducibility. So we at the library see many researchers come to us and they ask, how can I publish my data? Because the journal said I have to publish data. So that's the main reason they know this is a thing they could and should be doing. So I think this is really important. I just want to mention a specific example. This is the journal eLife. Some of you might know of it. It's a relatively new journal. It's fully open access. They also have a very nice policy for data sharing. They also have open peer review, meaning you can see what did the reviewers comment, how did the authors respond as part of the paper. And quite recently, they have what they call the executable research article. So I suggest you have a look at it. I just have a screenshot here. So they call it executable research article, meaning in the paper itself, in the online version itself, you can see the code. This is an example of the R script, I think, how they actually analyze their data. Of course, the data is also available. And then inline, in the browser, you can change parameters if you want to do slightly different analysis, run it, and see how it affects results. So you can actually see step by step what data did they use, how did they analyze this, and what are their conclusions. And I really hope they continue with this, because this is really using the tools we have to show research output in a different way. So we've talked about research data a lot earlier today, so I won't say much about this, but this is one of the important things we at the library and at the IT department do to support researchers, and that I think most universities also try to do and are working on to help support good science, open science, and reproducible science. So thinking about what tools do you have as a researcher for each step in this research data cycle. And I want to emphasize that open science and good data management, they go hand in hand. Open science doesn't mean everything should be open at every point in time. It means you manage and document and make your data and code fair. And then you open it up if it is correct to do so. So we talked about fair. I won't repeat that, but it's very important as open as possible, as closed as necessary, but it doesn't mean it shouldn't be fair. Fair is always the starting point. And if you have the time later, there's this lovely little video about what happens when data is not fair. So if you have the time, please have a look. And we also emphasize the reuse of data. So we now talk about opening up the data, but that's for a reason. We want to emphasize, try to reuse data in an interesting, exciting way. So I would encourage you all to become real parasites. So the Parasites Awards awards people that are reusing existing data in an interesting way and then publishing a paper about it. So be a proud parasite if you have interesting data to reanalyze. And I want to give an example of a research project that is more or less as open as you can get it. It's called the Open Source Malaria Project. So they are looking for new medicines, new formulas for medicines for or against malaria. And they are fully open in the sense that whoever wants can contribute. They have fully open notebooks. They share all the results and data as it's produced. Everyone can suggest, oh, we should maybe move in this direction. How about doing it like this? Let's change up the analysis. And all the results, all the possible medications they get, should not be patented. They are free for all to use and reuse. So this is the most open project I've heard about, where they are really every step of the way as open as possible. Of course, this is the extreme. We're not saying everyone should do this, but you can pick parts that seem interesting, making it your project as open as possible and as open as sensible and useful for you. So we've touched upon this earlier today. So why aren't more projects more open, or why is open science something we're still talking about if it's just good science? We've mentioned the infrastructure. You have to, of course, have good solutions, technical solutions. I don't think that's the main problem. I think skills, you're a bit uncertain how to do it. It's new. All these tools you have to know about can be a bit daunting. But maybe most importantly, the incentives. So why should you do all this when you, as a researcher, already are pressed for time? Why should you do all this when the only thing that counts, it seems like, is your publication? Why should you then publish the data? That won't help you get your next job. I think this is changing, but it will take a while. And we are used to, our culture and practice is, well, this is how we do it. We publish papers, and that's what counts. Process. Everything we do to make good science should be part of our output and part of what we're evaluated on. And of course, it takes time to learn new skills. It's also a bit scary, right? Publishing your code. If you have messy code, but that's so we shouldn't be afraid to put it out there, and then we at least can fix the mistakes if other people find them. And there are other initiatives to try to promote open science and transparent science. Many universities start to say, we want to show you to show us what you do as part of your open science practice before we hire you. This is also slow in uptake. Some institutions are better than others, but I think this also will have an impact in the long term. And as I mentioned before, also the funders, they do expect open science, transparent science, and reproducible science. I just want to mention a specific change. So you see this is dated April. So Horizon 2020, they quickly turned around and said, well, now if you get funding from us to research COVID-19, we have even higher expectations than before that you actually share your output. So of course, open access publications, that's always been an expectation. But they even say for research data you collect in these projects, within 30 days of collection, you should share it with the rest of the world. And now we're talking efficiency, and now we're talking sharing at a different level. And why shouldn't that be the default for at least some types of research? And there are, of course, a lot of interesting tools, platforms and collaborations that is nice to know about and can be really interesting to work with just a couple of examples. The Open Science Foundation, online tool for collaboration and sharing research data output. Also, the European Open Science Cloud is really good to know about. I won't go through this, but there are so many interesting, exciting tools out there working on open science. I suggest you have a look. Interesting and useful for me. So to sum up, open science is just science done well. You're kind of doing it already. It's not an either or, but it's a gradient and a process. It includes exciting tools, exciting collaborations and possibilities, and you're kind of already doing it. Thank you so much.
Thank you. Time for one quick question. So the first question up from Ellen was, which fields of science are doing the best and why?
Oh, yeah. Well, I can speculate a bit. I think you guys are doing pretty well. You're talking about standards. Not many other fields are doing that. Maybe the climate scientists and the physicists, they are pretty good. Yeah. Life sciences in general are pretty good. Yeah. That's a short answer.
Thank you. Well, we might go on to the next presenter. Thank you very much, Inge. Who are, I think there's plural. Yeah. So we have the status of the reproducibility crisis in the wildlife sciences. Can we, yeah. Click, stop sharing, okay. Technical difficulties, sorry. Excellent. So
Okay.
have we got Althea and John on the line? Yes. And we can, so for the people in the room here, we can see them up on the little TV screen there, and everybody else online should be seeing the speaker as well. Take it away. Thanks.
Okay, hi. Thank you, everyone, for being here and wanting to talk about open science. I especially want to thank the coordinators of this colloquium for inviting us to speak today. We are here to talk about computational reproducibility and drill down on that a little bit more, and specifically our study on the reproducibility of wildlife science studies. We have published this study with open access in the Journal of Wildlife Management, and we invite you to read our paper for more specifics about the methods and results. So in order to begin our presentation today, I want to define reproducibility as we applied it in our study, and then talk a little bit, or I guess now review a little bit of the phrase, the history of the phrase reproducibility crisis. So what is reproducibility? According to the National Academies of Sciences, Mathematics, and Engineering, reproducibility, and specifically computational reproducibility, means that an independent researcher can obtain consistent computational results using the same input data, computational steps, methods, code, and conditions of analysis as were published in an original study. So as we just saw, in 2016, a journalist at Nature polled over 1,500 scientists from around the globe and asked them, is there a reproducibility crisis in science? And the overwhelming majority said yes, with a whole 52% saying that there was that significant reproducibility crisis in science. So this paper and these conclusions led my colleagues and me to ask, is there a reproducibility crisis in the wildlife sciences specifically? So I will go through our methods that we use briefly and then share our results. So we wanted to determine the status of the reproducibility crisis in the wildlife sciences specifically. So we chose articles that were published in 2 of the leading journals of wildlife science in North America, the Journal of Wildlife Management and the Wildlife Society Bulletin. These are both journals that are published by the Wildlife Society, or as I may refer to it as TWS. So we randomly selected 80 total articles, 40 from each journal, that were published from June 2016 through June 2018. And we selected recent articles hoping that it would increase our ability to collect data from the authors. So for each article that we chose, we first determined if it was suitable for review, a requirement that was met simply by the article having quantitative results that we could try to reproduce. When an article was suitable for review, we began trying to find that paper's associated data on the internet, archived in supplemental materials. If we could not find the data online, then we emailed the corresponding authors up to 3 times. Ultimately, for each study that we were able to collect data for, we used the methods then to try to reproduce that study's results. And we rated our ability to reproduce the results on a 5-point scale and specifically determined if we could reproduce the specific numbers in the paper, the figures in the paper, and most importantly, the conclusions in the paper. So I want to note here that we may have been able to reproduce conclusions at a higher rate than we could reproduce specific numbers. So for instance, if we found that a correlation coefficient, for instance, was higher than the one that the authors had published, but that they were both still positive and significant, then the conclusion may have been reproduced even if the specific number was not. And then after that, a second reviewer from our team also ranked the reproducibility of each study, and we averaged across those reviewers. So let me introduce some of our main findings. So again, we started with 40 papers randomly selected from each journal. Of those 80 papers, we determined that 6 were unsuitable for review. We could not get data for 42 additional papers because it was not available online, and we could not get a hold of the corresponding authors even after 3 attempts. The authors from 11 papers opted out of sharing their data, citing reasons such as not having enough time to put the data together or being unable to track down the person responsible for sharing the data. And then another 2 authors never sent in their data even after agreeing to participate. Eventually, our deadline for submitting data passed, and we had to continue on to our study without their data. So that left us 19 studies for which we had the associated data. And of those 19 studies, 3 were completely not reproducible. They scored a 0. Another 3 were mostly not reproducible. They scored less than a 2.5 on our 5-point scale. 5 were mostly reproducible, scoring from a 3 to a 4.5 on our scale. And 8 were actually considered to be completely reproducible. So all in all, we concluded that 68% of the papers that we reviewed that for which we got data, wildlife scientists seem to be doing a great job reaching reproducibility. And I think that this is really promising. However, if we take a step back and look at the results from our original 74 papers that were actually eligible for review, we could only get data for 25 of those studies, making the actual reproducibility rate 19%. And this, I believe, is very alarming and indicates that we need to do a better job sharing our data. So I can't overstate this enough. It was extremely difficult to track down the data and the corresponding authors, even though these papers had been published at most 2 years prior to the start of our study. And I also want to point out that the data sharing is recommended in the author guidelines of both Journal of Wildlife Management and Wildlife Society Bulletin. And 2 premier editors-in-chief for the Wildlife Society have called out the importance of sharing data recently. So I think that this quote by former editor-in-chief of the Wildlife Society is especially potent. As glimpses of the end of my career flash in front of me, I have started to think more about what my legacy will truly be, my papers or the data that I leave behind. In fact, she goes on to point out that she wishes she could have done a better job sooner of archiving her data, and she ends by concluding that societies such as the Wildlife Society have a big role to play in making this paradigm shift a reality. Okay, so now we have a feel for our results, so I want to discuss 2 of the main obstacles to reproducibility as I see them. The first obstacle is being able to write your methods that are clear and thorough, that document the entire process that you took to reach your conclusions. And we found this commonly in our analysis, right? That well-documented code, well-written methods, thoroughly described data processing steps all increase the chance that we could reproduce an author's results. So on the other hand, those that didn't take these steps and whose papers scored lower on our scale, I think could have fairly easily overcome these obstacles, especially now in a world, as we're seeing through the whole Living Norway Symposium, that there are many, many resources out there ready for us to do a better job making our coding and analysis reproducible. So we've been talking about this all day. There's articles on pre-registering your analysis, tips and tricks for writing code that's easy to follow, tips and tricks on how to archive data, and so on and so forth. And honestly, it takes a little getting used to, but once you start approaching research with a reproducible standpoint, it's not very hard and can even become habitual. So as this researcher pointed out, reproducibility is like brushing your teeth. Once you learn it, it becomes habit. So I want to move on to actually what I think is the more challenging obstacle: sharing raw data openly and freely and with trust in each other as fellow scientists. Since we began our study in 2018, there's been a little bit of a sea change in what I view as our willingness to share data freely and without reservation. So for instance, more and more journals are including mandatory data sharing policies, such as the one at +1 that we saw in the previous talk. Archiving frameworks are becoming easier to use and more prevalent. And I think this is all good because, as Dr. Miyakawa at the Molecular Brain stated, and we just saw in the previous talk, no raw data, no science. So to put the title of his paper, 2020 paper, in context, I'd like to share a quote from that paper. Making raw data openly available is not only important for reuse and data mining, but also for simply confirming that the results presented in the paper are truly based on actual data. And he shared in this paper that out of 170 articles that he has managed since 2017 as editor-in-chief of Molecular Brain, he sent back 41 studies with a revised before review status and a specific request for them to share their raw data. Of those 41 studies, 21 withdrew their papers without even sending in data at all. Of the other 20 studies, 19 were rejected because the raw data that they provided were insufficient to support the results in their paper, and only one was published. So he continues on in his paper to emphasize the importance of sharing data, and that from his viewpoint, those authors that withdrew their papers could easily have been suspected of mishandling their data in an effort to report favorable findings. Of course, he couldn't prove this, but I do think that it shows a much higher level of reliability when we are willing to share our data. So in light of the difficulty for many authors to get data when conducting tests of reproducibility, because we were not alone in finding it difficult to get data, as can be seen in many other similar studies from different fields, Dr. Marcus Manufo and others have recently founded the UK Reproducibility Network. And one of their main missions is to create a paradigm shift related to sharing data and open science. And specifically, they state that to change our willingness to openly share data, we may have to change the fundamental incentive structures with which researchers operate. That is to say, the underlying obstacle to free and open sharing in science is directly related to the pressure to publish novel results, and that we won't see major paradigm shifts until we tackle that underlying culture. So in addition to my usual recommendations to always write better code and organize your data to increase reproducibility, I think it's even more important to better tackle these underlying issues. My final call to action. So first, I hope that we can normalize the sharing of data and code. If we all write our code from the beginning with sharing in mind, this process, again, just becomes habitual. Secondly, let's begin requesting raw data when conducting peer review. So if we're confused about how an analysis was conducted, or for instance, how random effects were handled, in addition to asking for them to clarify that information in their paper, we also could start asking for the data in the code to learn more about how they analyze their data. Third, let's encourage journals to incentivize data sharing. I think we're going to hear more about this in the next talk. Perhaps there could be small reductions in page costs or some recognition for those of us that share our data readily. Fourth, let's encourage journals to publish sound replications of previous papers. regardless of the results. So let's break down the pressure to publish novel results and instead embrace the importance of building off of previous work. Let's view papers that agree with previous findings as the building blocks to strengthen our understanding of science. And then fifth, I'd really like to see us all doing a better job choosing a stable email address as corresponding authors and using things such as ResearchGate or ORCID to maintain a trackable online presence. As we found in our study, it's so important that we all respond to PDF requests and emails, and let's just work together. And then finally, we can never forget how important it is to train each other in better, more reproducible practices. Think about the legacy of your data and the legacy of future research, and let's all try to leave the scientific world a better place than we inherited it. So I want to leave you with this quote by Paul Krugman, who's the editor-in-chief for Journal of Wildlife Management. Publish all of the relevant results from your research and archive your data for future use. When you notice your data in publications by others, know that you had an additional part in advancing our profession. And I want to thank our large team of collaborators for this study, because I couldn't have done this without everyone pitching in and helping me try to reproduce all the studies. And with that, I want to close and hopefully have time for a couple questions.
Thank you so much. It's very weird asking questions for a person who's not in the room, but we have some on Slack. So one of the first questions was, have you got any idea about what, why you couldn't find some of the authors?
Yes, so we had a little bit of kind of an informal follow-up with some of the authors that people knew kind of professionally. So all of the emails came from me originally, asking for the data, and we did a little bit of a kind of informal follow-up. And in a lot of cases, it seemed like there was that time was a really big factor. And so if they weren't going to get credit for sharing the results, then why would they take the time to put their data together, right? And another big obstacle that we heard, I think several times, was just people not like changing affiliations, and or the person who had the data originally maybe finishing their postdoc and moving on to a new institution and kind of losing the trail. And I think that was kind of surprising, since we started our study in 2018 and only went back to 2016 papers. So it was surprising to us how quickly so many people lost their trail of correspondence.
Yes, and I guess ORCID is one thing, but if we lose these people out of academia completely, then yeah, I guess that doesn't follow them.
Yeah.
We have a couple of other questions also on the time it takes to do these things, and the time it takes to share data, and the time it takes to peer review data. And even when you request to peer review the code and the data, do you get extra time to peer review this? And yeah.
That's a really good question. I think that's something that the journals are going to need to consider as data peer review becomes more and more common. That comes along with more time to do that review, because it does take a long time to work through someone else's data and try to get a feel for the data in the first place, and then get it processed and analyzed. And that is all, of course, made a lot easier if data are analyzed with open source technologies and command line based analysis programs. We really struggled when people used drop-down menu style analysis programs to try to reproduce those results.
And have we got time for one more question? Yep. So this one might be for both you and the next speaker. The oft quoted excuse of not providing peer reviewers with raw data is the fear, unfounded or otherwise, that the authors will be reviewed by a bad faith actor that and publishes the data themselves? Do we need to quell these fears through open review to encourage submission of data and code?
Yeah, I think that the so you're speaking to kind of the fear and mistrust, and I think that that is really so strongly built into that underlying idea of the pressure to publish, and that is our currency. And so we're so afraid that if we lose our ability to publish our own data and the credit that goes along with that, I like to have better faith in other people and in our fellow scientists, and I just have hope that that paradigm is starting to change, and that, again, as the previous speaker said, we are all doing open science to some regard, and it's just going to become more and more commonplace.
Maybe one other quick point there is if you publish a preprint, then you can demonstrate that you were ahead of you were the first one to publish. You're responsible for those data and the methods and the results to get out ahead of the game and avoid that concern.
Yeah, thank you, John.
Great point. Okay, so we probably should move on to the next presentation. Yeah, another online one from Emily. Hello, Emily. Is Emily perhaps muted? Or...
Hi, hello, can you hear me?
Yes, we can hear you
Can
now.
you hear me?
Yeah.
Okay, great.
I think if you could just pop it onto share mode. Yep, perfect.
Yeah,
Thank you.
I'm just... Okay. So hi everyone, thanks very much for inviting me to talk today. It's really good to be here. My name's Emily, I'm one of the managing editors from the British Ecological Society. So I look after the journal development for several of our journals, and I also work on some open data projects, and hopefully in this talk I can speak to a few of the things that have been mentioned earlier in the session. First, I just wanted to tell you a little bit about the British Ecological Society. So we're the largest ecological society in Europe, and we've got over 6,000 members around the world. We pride ourselves really on being a very kind of open and welcoming society, and I'll tell you a little bit more about kind of membership of the society at the end of the talk. So a bit about what we do. So we give out over £350,000 in grant funding every year to members of our society. We have a big international conference every year. This year it's going to be completely online. And we hope that that will mean lots more people than usual can join us. We also have kind of several special interest groups for various disciplines, sub-disciplines in ecology. And these are a really great forum for collaboration and knowledge exchange. We also do a lot of policy work and education work for early career researchers. And then we have our publications programme. So these are our journals. So we've got 5 really well-established journals in ecology and evolution. 2 of them launched just in the last couple of years. So People and Nature is our new interdisciplinary journal. And we also have Ecological Solutions and Evidence, which is our newest journal. And that's part of a new resource, Applied Ecology Resources, which is a database of grey literature, summaries and open access articles. And these really try and promote evidence-based decision-making in ecological management. So I guess hopefully in this talk I'll show you that journals are already doing quite a lot to support open data and discuss a bit about what we can do in the future. But I guess before we start, I'd really like to ask you guys a bit of a question. So what is it that you would like to see journals doing to support open data? I guess we've spoken a little bit about that in the session up to now, but as you're in your position as kind of authors and users of data, it'd be really great to hear from you. So if you could put any comments about that in the Slack chat, that'd be great. So to tell you a little bit about open data at the BES, so in before 2011, although we talked a lot about open data, we didn't particularly have a set policy on it. And then in 2011, we signed up to the Joint Data Archiving Policy. This was with a lot of other journals in ecology and evolution that signed up at the same time. And I think we decided at that point that we needed to start quite slowly and get community buy-in. So at that point, this wasn't a mandate, it was just that authors were actively encouraged to archive the data, supporting their articles. And then from 2014 onwards, authors have been required to archive data as a condition of publication. So data need to be freely and permanently available. And so I guess what do we mean by freely and permanently available? Well, they need to be free to download without any kind of barrier. So that means that available on request isn't appropriate. And there needs to be some kind of guarantee of permanence, like a DOI. So that means that putting it on an author website also doesn't work for us. So sufficient data also needs to be archived so that the results can be reproduced in full. And we also ask that code is archived for software and theoretical papers. Well, but what about my special situation, people often say? Well, it's not quite as simple as that. So we allow a 1-year embargo if it's supported by the repository, and that's no questions asked. And then longer embargoes are allowed at the editor's discretion. So the most common reasons for this is often for kind of PhD students who we allow to kind of have their data for the whole time they're doing their PhD until they've written their thesis. And then the biggest, our favourite reason really is to try and avoid breaking up big data sets. So if authors need a bit more time to use their data, and that would allow them to archive a full data set rather than kind of fragmenting it for the publication, then we think that that is a good reason to allow a longer embargo period. Complete waivers of the policy are also allowed sometimes, so that's often generally for if data are identifiable for humans, or if it would show the location of endangered species. The other main reason that we can waive the policy is that the data aren't the authors in the first place. So in this case, we ask that the authors have asked the data owners to archive the data, but if the data owners say no, there's not a great deal that we can do about that, really. So reasons that we don't allow people a waiver of the policy is that, well, the data are already in supporting information. We don't allow data to be archived in supporting information. It's less kind of findable, and it's kind of not as good as a proper repository. And often people just think that they should be allowed to keep the data because it's theirs. In our experience, there is a bit of a fear of being scooped. This tends really to be much more of a fear of the thing happening than it ever actually happening. I think it happens in real life very, very rarely, but it is kind of a fear that we, I think, need to address as a community. So how do the BES journals manage this process in practice? Well, at submission, authors have to agree to our policy, and they have to say where they will archive the data. If this information is missing or incorrect, it's queried by the editorial office. And then at the acceptance stage, data has to be archived before we can finally accept the paper. So the editorial office check that the data are there and that files can be opened, and they also check that the repository meets the requirements of our policy. This does take a bit of time, but it is manageable, and I think generally the most time-consuming thing is checking that new repositories that we haven't come across before, that they meet our policy. I guess it's important to note that academic editors are not often involved in this process. They tend to only get involved if there are kind of tricky cases, where there's some kind of issue with the data or with the repository. In general, this is handled by the editorial office. So how do we support our authors to meet this policy? Well, I think the main thing that we do really is to cover the cost of data archiving in Dryad. Dryad's the most used repository for ecology and evolution journals, and it does provide enhanced curation for data. So we do support Dryad, but we also allow authors to use any repository they choose as long as it meets our standards. We provide a lot of training for early career researchers in this, and in the form of workshops, and also in the form of our guide to data management, and this includes lots of tips and checklists for those new to the process of data management. And then for our authors, when they're submitting, we provide quite a lot of detailed guidance within our policy. It's not just a kind of a couple of line policy. It also includes a long kind of checklist of how to archive data, and also a list of appropriate repositories that people can use. So what about other journals in ecology and evolution? So we track around 25 journals, and of those, all of them have some kind of policy on data archiving. Around half of them encourage data archiving, and half of them require it as a condition of publication. 4 of them that we look at ask for data at an earlier point than at acceptance, but often that's not compulsory to do that. I think it's worth noting that all the journals have a policy, and the number that mandate data archiving is increasing. So in ecology and evolution, there are a variety of repositories that are used. As I said before, Dryad is the most popular, followed by Zenodo, and then followed by Figshare. Zenodo has grown a lot in the past few years, and I think will continue to do so. Institutional repositories are becoming increasingly important. Institutions are now starting to mandate use of their own repositories, and that's for that reason they're growing. And then there are lots of specific repositories for certain types of data, and often these are the best place for a particular kind of data. I think it's worth noting that I think there'll always be a lot of different repositories in ecology and evolution, and that's because there are a lot of different kinds of data, and that's absolutely fine as long as they kind of meet certain minimum requirements, I think. And in general, the number of data sets archived is growing year on year. So last year we conducted a survey of BES authors and reviewers, and we found that nearly 90% of respondents support the requirement to archive data, and actually close to a third of them think that data should be archived sooner. 13% think data shouldn't be archived, or archives should be longer, and most of these people are concerned with being scooped rather than actually disagreeing on principle with data archiving, and actually we found that only 3% disagree on principle with archiving data. In general, I think there's excellent buy-in from the community, and they're much more comfortable with data archiving as the norm than when the policy was first introduced. And I think it's also promising to see that so many would like to see data potentially archived sooner than at acceptance. So I think the question of why people archive data is an interesting one. Most authors do so because they see the value in it for the community, so because they want to make data or code available for others, because they want to ensure that results can be reproduced, or making data and code available helps to understand the article. But I think it's really important to note that 64% archive data because it's required by the journal. We'll come back to this point. I think really important in this area. So compliance with the current policy is excellent. At the BAS, we see close to 100% compliance with our policy, and we have over 3,500 data sets available. We put a lot of time and kind of effort into policing this, so it's perhaps unsurprising that we get such high levels of compliance. And I guess going back to what I said before, it is mandated by the journal, and so people do what they're told, essentially. And yeah, just another example of the fact that mandates work here. Most journals do have a policy on data, and only some have a mandate. So looking at the journal Animal Behaviour, they have a policy of encouraging data sharing, but they don't require it. And in 2019, they had 24 data sets on Dryad and 7 in Mendeley data. If you contrast that with behavioural ecology, who require data to be archived as a condition of publication, in 2019, they had 543 data sets on Dryad. And clearly that shows that these mandates really do make a difference. So as I've discussed, journals do a lot to support good data practices, but there are some issues. Data quality is often variable, often metadata is poor quality or missing entirely, often the description of how to use the data isn't particularly useful for those who aren't the authors. And sometimes data sets are actually incomplete, which would mean that the results aren't in fact reproducible. Interoperability is often a problem too. Authors aren't asked to adhere to any kind of consistent standards when they're depositing data, and data are often in kind of different file formats. So this means meta-analyses are particularly difficult or sometimes not possible. And I guess the major point is that journal data policies are really there to promote replication of the work that the journals publish. They're not there really to support reuse of data. This is kind of a secondary thing at the minute. And this means that data are often fragmented because they're archived to go with the particular paper in question. And also because authors are concerned about being scooped, they try and archive the kind of minimum amount of data necessary. And this is generally fine for replication, but it's not efficient for reuse or for answering new questions. It's not all doom and gloom. There are more data available than ever. We've come a really long way in the last 10 years from a situation where pretty much no data were available to the point where at the BAS journals, as I said, over 3,500 data sets are available. Across ecology and evolution, lots of papers do now have some useful supporting data available, and this does mean that reproducibility has improved. It also means that any errors are easier to spot, and it's easier to correct the publication record than it ever has been. Archiving data is really now considered the norm in ecology and evolution, and as I said before, only 3% of the people we surveyed disagreed with data archiving on principle. So hopefully we've seen that that journals already do a lot to support these good practices, but what more can we do? So it seems clear to me that it would be useful to have some kind of standards for data sharing across the discipline. But if we introduce these, they would need to be practical for authors, otherwise they wouldn't be used. And I think journals will need to work across the community with other journals and researchers to figure out what these standards should be. I think funders are increasingly requiring clear data management plans now, and they're starting to follow up and check that data are archived appropriately. So if journals can align with funder policies and any mandates that they introduce, journal policies are more likely to be well received, I think. And I guess we've also seen that mandates work, so any policy changes would need to be requirements from a journal end, I think. But in order to get to a point where we get community buy-in, I think we will need to work together. So an issue is that introducing new standards under current journal processes, as I said before, currently these are checked by the editorial office, and if we introduce standards for data quality, that wouldn't be possible. So we would need to police it somehow, and I think in order to do that, we would probably need to make authors make data available sooner. There is an issue with rejection rates in this situation. Most BAS journals have very high rejection rates, and so that would mean that some authors were archiving data for their paper then to go on to be rejected. I guess you would hope that eventually it gets published somewhere, and that's still useful for the community. I guess we'd also need to involve journal editors more often and sooner, and similarly with reviewers. This could potentially be a lot of work, as the last speaker suggested. We'd need to make sure this was practical for everyone involved. Clearly, I don't have all of the answers here, but it seems clear that any improvements that journals make need to be based on collaboration with researchers, societies, possibly funders and repositories as well. It feels like in order to get buy-in, we'd need to facilitate a culture shift from data being an added extra to papers to being an integral part of the published output, and it just being what you do when you're writing up a paper. I guess these changes sound like they're quite big, but as I said before, we've come a really long way in this area in the last 10 years or so. Data archiving used to be quite scary for a lot of people, and I guess now it's much more considered the norm for ecology and evolution. I think, as with any new changes to policy, and as with our previous changes in this area, they'll need to be introduced gradually, and the benefits will need to be clearly communicated. Consistency between journals and even between journals and funders, I think, will help with this. And I think, as I said, any changes will eventually need to be mandated to work. At the BAS, we're really keen to work on this in practice and not just in theory. We've started having discussions with researchers and with other societies to do some of this work, and we'd be really keen to hear from anybody who's interested in being involved in this, I think. And with that, I'll say thanks very much and take some questions. I'll also just put this slide here about BAS membership.
Thank you. So we have a number of questions. We haven't got time to go to many of them, but one of the first ones was, do BAS journals support open peer review, i.e. peer reviews that are published alongside the paper with credit, etc.?
So we're actually, a couple of our journals are doing a trial on open peer review at the moment, where authors can opt in to have open peer review, and if they do that, then all the peer review reports are published on Publons. Authors can choose to have their name associated with them or not. So yeah, I think it's something that we're trialing, and we'll see how it works.
And one from Joe, which is slightly long, so I'll try and paraphrase. So he has a worry that the suggestion that moving towards data papers will prevent some of these data going into more databases and potentially locking up this data and the metadata into published papers that aren't open and free. Can you comment on how data papers can be more free and more linked to some of these open databases?
Yeah, absolutely. So data papers is an interesting one. I think data papers make most sense when they're open access, but I think even when the paper itself isn't open access, most data journals or data papers will require the actual data and some description of it to be made available in a publicly available repository. So hopefully that would solve that problem.
Sorry, there's lots of comments on the stream as well.
Sorry, that's my fault.
No. Well, it's your fault for having an interesting talk, I guess. Why only code for theoretical papers? I'm not quite sure.
Oh yeah, so it's not just for theoretical papers, I guess. It's for, for theoretical papers, we absolutely have to have the code available. For any other kind of papers, we need sufficient data available for results to be reproduced, and often that will involve people archiving their scripts that they use to analyse the data as well. Sometimes people don't do that, and it's still possible to reproduce results without that. But it does help, but it's not mandatory for standard data papers.
Excellent. So I think with that, there were more comments rather than questions, which were excellent. I would like to thank everybody who spoke for wonderful talks, and we can have a coffee break.
Hallo. Der, ja. Kommer han opp? Hvordan Hvordan er lydnivået der på Hallo. Hører du? Ja.
Får dere til å slå av lyden så det ikke kommer ut på streamen?
Gavin
Okay.
Stewart. Gavin Stewart, yes, there you are.
Ok, nå
I
prater jeg.
will...
Høres det greit
Hæ?
ut der ute? Hallo, 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 hallo.
last session for today, moving on to the topics of how open science and fair data can be used in the science policy interface. We have a slight change in the program in terms of the order of the speakers. So the first speaker will be Cecilia, and perhaps you can already start sharing your screen, Cecilia, while I introduce you. Cecilia Denby, she's the director of the Norwegian Scientific Committee for Food and Environment. And she will be talking about how open scientific knowledge synthesis can be used and her experiences from the Norwegian Scientific Committee for Food and Environment.
Yes, can you hear me?
We can hear you, but we don't see your screen.
You don't see the screen.
Maybe
I'm
it's coming.
working on the sharing.
It's very exciting to wait and see what
It's
pops up.
always exciting. Do you see it now?
No, we don't. I don't really have any tips and tricks, so I'm just waiting and
No,
hoping.
we have just tested it 2 minutes ago and it worked, but
Then it probably will work again. Looks like
let's
all
hope
the
so. We
people-
lost the sound of the other participant, and then now we lost the screen from me, so that's typical.
Looks like all the people in the technical committee behind there are working very hard.
Oh, it is, yeah. I'm sorry about that.
This is the moment where I should be a really fun person who could tell jokes or something, but I'm not.
I'm not telling any jokes. Do you have the microphone here? Can I borrow this one, perhaps? Cecilie?
Hallo?
Hei. Nå er det bare å dele på samme måte som i sted. Vi hadde litt problemer med videoen.
Nå hører jeg ingenting. Can't hear anything.
Hører du meg nå?
Ja, nå hører jeg.
Ja, det er bare å dele skjermen på samme måte som i sted. Og så hadde vi litt problemer med videoen som kom og gikk fra Cecilie. Så hvis vi ikke får med video, så er det sånn det. Det er noen bredbåndsproblemer.
Jeg driver og deler her. Den står og på en måte jobber, men
Prøv å kansellere delinga og kjøre en gang til.
Skal vi se. Ja, nå står den bare og jobber, så jeg får ikke valgt noen ting. Jeg trykker på escape. Det går jo heller ikke.
This is the perfect moment for discussion on Slack. Something else.
Det var da veldig uheldig da.
Little explanation in English here. We have two presenters with a little bit of connectivity problems now. So that was why we had to change the order. Might have to do that again.
Yeah, it says that Google Chrome doesn't reply from this side here now, and it's sort of working on this sharing thing. So should I just go out of the whole thing and connect again?
Yes, I think that's a good idea.
Ja, ok.
Skal vi skifte en gang til da, for her er det Reiner Krog. Han står klar.
Let's test if it works, and then we just take whoever speaker can have their slides presented at the moment. We can always change the order back again later, I guess. So should we ask if Reiner could?
Yes, Cecilia, are you with us? Reiner, are you there?
Yes, I'm here.
Okay, promising. How would you feel like presenting right now, directly?
Oh, it looks like something is happening here.
Yeah. Wouldn't be a problem.
Okay,
Okay.
then if it's okay with you, then let's just welcome Reiner. Skal du ha den?
Nei, jeg har allerede den her. Okay, then we go for another change again, and we give the word to Reiner Krug, who is the lead of Data Management Working Group of the IPBES Task Force on Knowledge on Data. So please go ahead, Reiner.
Yes. First of all, I would like to thank you for the invitation to speak at this colloquium. I must really say it's very interesting and very thought-provoking. I'm going to talk about IPBES and implementation of a data management policy in IPBES, of the lessons we learned from implementing that data management policy, and further developments.
Reiner, excuse me, can I just interrupt you a bit? It seems like you have a different view, because we can see the notes and the next slide and so on. We don't have the full screen of the... There, perfect, thank you. Please go on.
So I'm going to talk about why IPBES needs a data management policy and lessons learned from the development of this data management policy and further developments for this data management policy. But before I jump into this discussion, I think I probably should introduce IPBES, what IPBES is doing, and why it plays off kind of a special role in that whole development of the data management policies. Well, IPBES is an institution which is sitting in the science policy interface. It is, and let me just read that one out, that's such a nice sentence, Intergovernmental Science Policy Platform on Biodiversity and Ecosystem Services is an independent intergovernmental body established by states to strengthen the science policy interface for biodiversity and ecosystem services, for the conservation and sustainable use of biodiversity, long-term human well-being, and sustainable development. So the important parts in this description are: it is independent, it is an intergovernmental body established by states and working on the science policy interface, including sustainable use of biodiversity, long-term human well-being. So the question comes, why does an organization which is working on the science policy interface need a data management policy? Why is it not moving to the next slide? There we are here. Okay. So why does IPBES need a data management policy? To understand this, we have to go a little bit closer into how IPBES is working and what IPBES is actually doing. IPBES consists of 5 different work packages. There is first of one, the assessment. In this assessment work package, they are creating assessments of existing knowledge. These ones are used for policy support by compiling these assessments, which are sometimes around 1,000 pages, into shorter documents, which are then used in supporting policy. They are also working in the area of capacity building, of knowledge generation, in other words, of filling of gaps in the knowledge identified during the assessments, and obviously communication and outreach. So it is essential that IPBES has a policy relevance, that it is credible, and that the assessments are by the different governments in different places. So we have knowledge and data which exists outside of IPBES, and it is important to know, which is essential, that IPBES is not generating data. IPBES is not going out collecting data. It is using the existing data. So the existing data is used by IPBES in the process of creating the assessments. And to create these assessments of the existing data, the existing data is integrated and then used to compile these assessments. So what is IPBES doing, and why does it need a data management policy? That goes around the points of credibility. If you have a closed process where you're using existing data and this process is not reproducible, you cannot find the source data. That whole process and all the results in the assessments are not credible. So IPBES is striving towards a higher credibility, a higher transparency, higher reproducibility of all the assessments, and not only the whole assessments, but each single table map in the assessments, which is compiled using existing knowledge and data. The aims are a little bit more detailed, that all the knowledge generated during this integration is archived for the foreseeable future, that each individual piece can be identified through DOIs, that for this process of the integration, open source tools are used for the data gathering and for analysis, that well-defined metadata schemes and standards are used, that code and generated data, so in other words, the code is used to generate the data. The data could be maps, for example, compiled by using existing records of species of all different kinds of layers. This data is available in interoperable open formats. It is reproducible and searchable and findable from IPBES website and from other data portals. After these sessions we had before this morning, this should sound quite familiar to you. And it essentially boils down to fair data usage standards. If I would have known how often fair was used this morning, I could skip the next slide, but still I think it's a nice slide. So fair data implies that the data is findable, that it is accessible, that it is interoperable, and that it is reproducible. These are the 4 pillars which the data management policy wanted to achieve within IPBES. For that data management policy to be developed, that whole process started in 2019 in a meeting of the Knowledge and Data Task Force of IPBES, during which there was lots of talk about these different 4 pillars of the fair data management. And it boiled down that it was said we need a data management policy for IPBES. And then the process started, it was drafted and developed, and finally, about half a year later, the data management policy was approved. And it is available on Zenodo. You can access it. It is at the moment in version 1.0, but we are working on a few changes for the next version, and I will come to these points later on. The objectives of the data management policy are, in the first sense, to make all data fair, that all data, all code which is used within IPBES, within the assessments, is fair, and all implications of data being fair. Then to assist experts in making their outputs fair. Experts are coming into the assessments and they have never worked with IPBES before. They are probably not familiar with the data management policy, probably not even with FAIR in the worst case scenario. And the data management policy specifies structures and approaches to assist these experts in making the data they are generating, the code they are using within these assessments fair. There policy wants to include or foster the development of data management plans so that data management is planned when the assessment is starting. Providing for long-term storage, not for providing own long-term storage repositories, but providing guidelines for long-term storage. It includes sections about the tool selection for the assessments, and they should be open source. There's a heavy push towards open source tools within the assessments, but also in the whole of IPBES. The experts should follow the data management policy and, in other words, the fair data management principles. And the last point is that the data management policy states that the use of open source tools should be promoted within IPBES and the fair data management principles as well. Now, coming to the second point. Which lessons did we learn from the development of the data management policy? Well, the lessons learned are, first, the science policy interface needs a data management policy. Even if it does not look obvious when you're looking at it first, the science policy interface is using data, it is integrating data, it is drawing conclusions out of data, which are usually not conclusions where somebody's sitting behind a desk, oh, that data could tell me that. It is based on statistical analysis. And all this goes towards a data management policy. The second point is that all different aspects within this data management policy profit from, within IPBES or in that organization, profit from the data management policy. The positive impact is on capacity building. It is on outreach. If you have open data sets, if you can demonstrate how the results were obtained, you can use that in outreach. You can use that in capacity building, which is a huge area where we're going to focus on much, much more in the next coming years. The third point, a very important one, data is not data. If you're talking with an ecologist about data, the image they have about data is completely different from somebody who is working at the science policy interface, or even more extreme, somebody who is working with indigenous and local knowledge. All these different kinds of data, sociological data, let's call them hard data, and that's not judgmental, hard ecological data, indigenous knowledge, they are forming an integral part of the work of IPBES and of many, many other science policy interfaces. And the data management policy needs to cater for that, which makes the data management policy for these policy interfaces quite different to the ones for other scientific institutions. And consequently, the requirements of traditional data management policy need to be adapted to fit to these different kind of requirements and different kind of data. Coming to the last point, further developments for the data management policy. Well, there are a few areas where the first one is most definitely that the quality of the assessments must not be compromised. If any of these policies lead to a compromise in the quality of the assessments within IPBES, this cannot be justified to the governments who mandated IPBES in the work of IPBES. So the quality must not be compromised. The second point is something which we just realized a few months back, that data management plans do not work in the context of assessments. Assessments are dynamic. Data is gathered. The data is not available initially. Nobody has an overview of the data when the actual work starts for the assessment, but it is changing over time. So you cannot develop a data management plan in advance. we decided to move towards a data management report, which is developed in parallel to the actual assessments and includes all the information which is usually in the data management plan and some more. The next part is an area which I personally consider as extremely important, is to include indigenous and local knowledge more in data management policies. That's a long topic, and one could talk a long time over this, but I just want to point out two aspects. It's a completely different kind of data. Indigenous and local knowledge is given in stories. You have drawings, you have dances, you have songs, which are essential and contain lots of important information. And the second area is there's, in addition to FAIR, there are the CARE principle for indigenous data governance, which are talking about use of the data, benefit sharing, and so on. They are very nice, very interesting principles, but there might be possible contradictions with FAIR. And this has to be assessed in detail, and that's one of the big chunks of work which we are trying to address in the next year. The last one is data sources. It was already discussed. At the moment, it is required that data is findable, but not necessarily accessible. But we are trying to move more towards findable and accessible, and trying to avoid closed databases, closed data sources. And as a last point, I don't think if I mentioned that one already, the quality must not be compromised. So we cannot exclude data sources if the quality of the final product of the assessment is compromised by excluding that data source. I would like to end with that picture, with that image, which is the Knowledge and Data Task Force of IPBES. And without all of these people, that whole development of the data management policy, implementation of the data management policy would not have been possible. Thank you. Yeah. What do we have to do now?
There, I think my microphone is on. Thank you, Rainer. We are running a bit late, but I was thinking if perhaps Gavin could test if he is able to share his screen, and meanwhile I can ask you one question, Rainer. I'll pick one of the questions from Slack. What would you say is generally the main reason for data gaps when conducting the assessments in IPBES? Is it lack of data, lack of access to existing data, or is it the pure quality or documentation of the open data?
Obviously, I don't think you will find any scientist who says that we don't have a lack of data. So everybody needs data, everybody wants to have data. But mainly in the context of IPBES, the gaps are, they're not because of something, but they are identified. Only if you're doing a large-scale assessments of a certain topic, you will see certain things where you realize that information is missing. So it is really, you're just doing that assessments and you're identifying these gaps. There is not that somebody, that there's no interest in addressing these gaps, but one has to find the gap before one can close it.
Thank you. I think that we are trying to get the next presentation from this laptop here in-house instead of streaming it. But I guess you are with us with sound at least, Gavin, right? Can you hear us?
You hear me?
Yes, now we can hear you.
Right.
So I can introduce you while we are getting the presentation up on the screen. Maybe we have to use this here to share it. Let's see. Yeah, there it is. So Gavin Stewart is the director at the Evidence Synthesis Lab at Newcastle University, and he will talking about how we can go from bad science and science fiction to evidence-based X, how we can fix the world with evidence synthesis. So who's in charge of, you're in charge? Okay. So please go ahead, Gavin.
Okay, doke. Thanks very much for inviting me to talk to you. I think the first assumption that I'm going to make in this talk is that evidence synthesis is important and valuable in the generic methods that we use to accumulate knowledge irrespective of discipline. And this isn't an issue about replication per se. We're not talking here about is a study reliable? We'll come to that later. And that this method of knowledge acquisition is really important in disciplines like ecology, where there's an awful lot of heterogeneity. If we're ever going to understand that, you need to look at more than one study. So I'm kind of assuming before I start that looking at more than one study is what you want to do if you want to reliably inform policy. Matt, next slide, bud. Okay, so let's look at this to start off with from the perspective of a practitioner. And let's say that you work with salmon, and you work with an organisation that's given you a load of money to increase the abundance of salmonids in a catchment. What do you do? Where do you start? Next slide, Matt. Okay, well, the first thing you're going to do is you're going to go and get some information. You might talk to your colleagues, you might talk to scientists. Apparently Google thinks this is what scientists look like. I think ecologists sometimes look a little different. You might read some books about integrated watershed management. And you're almost certainly going to do some kind of searching of the literature. Next slide, Matt. If you go off and you do that whole systematic review thing and spend, a year of your life searching through the literature exhaustively, you'll end up with a list of studies like this. And they've looked at lots of different interventions, and there's lots of literature on those interventions, and they measure lots of different outcomes. But it's all basically trying to get at the question of, how do I get more salmon in my river? Or trout, yeah, salmonids. Next slide, Matt. Now, if you read the author's conclusions, then everything works and it's all wonderful. If you read the abstracts, everything works and it's all wonderful. If you get a machine to machine learn or to do sentiment analysis or topic modeling, everything's wonderful. But if you use those papers as a source of evidence and you actually try and look at them deriving effect estimates, so you're literally just using them to recreate the data that's contained within them, then you end up with a very different picture. Next slide, Matt. And unfortunately, you enter a horrible world of pain, which I'm going to illustrate with this this particular manuscript. Apologies to the authors. Next slide, Matt. Okay, so effectively with this paper, the researcher degrees of freedom are almost infinite. Okay, they measure two different outcomes and they measure them in different ways. They're looking at multiple interventions, so multiple ways of trying to get more fish in the river. 7 or 8 different ways of doing that. They're looking at those 7 or 8 different things in multiple different contexts, so different river flows and all the rest of it. And they're looking them at multiple time points, and on top of all of that, they're using 2 sampling methods. Okay, so when you pick this paper up and read it, you just get page after page after page of graphs and data, and it's really difficult to even figure out, reading the methods, exactly what they've done and how they've done it and when. And you read through all of this thing and you try and make sense of it all, and you can't, basically. It's very difficult to make sense of it. But if you read the abstract, that's fine. It's perfectly clear these things work to increase the abundance of salmonids. And of course, those conclusions are based on one statistically significant result. They don't look at the multitude of all of these results and all of this data. Okay, next slide, Matt. Okay, so let's get back to our poor practitioner. Okay. They want to do the right thing. They want to spend this money in the right way. They want to make sure that they're being effective, that they're using evidence-based practice. They've consulted their colleagues and books. They've visited an information specialist to make sure that they've got all of the literature, not a biased subsect of it. They've waited a month for the results. They've employed research assistants and a statistician and waited another month for them to get back to them. Maybe they've employed a meta-analyst who's looked at all of these studies and combined them in some way. What can we tell them at the end of all of that? We can tell them very little most of the time. Okay, the evidence base is depauperate, the evidence base is incoherent and inconsistent. And then you think, how much research effort have we wasted? All of those studies, and we can't really say what's going on. Okay, and we can't say what's going on because our data isn't fair. And that's the main problem. So at this point, our practitioner is crying, I'm crying, and the funders are crying. Next slide, Matt. And of course, this applies at a much broader scale. You know, if we come away from salmonid management and specific interventions, this applies to whip bears or to thinking about climate change, life below water, life on land. These big societal issues, global food security, all the rest of it. Next slide, Matt. And what we've seen there is this problem of the replication crisis, the problem that, you know, studies don't replicate because they're poorly executed and poorly designed and selectively reported. Next slide, Matt. Okay, we exacerbate all of those problems with our use of p-values. You know, we all listen to Fisher and we should have listened to Gossett. So we conflate strength of evidence and statistical significance all the time. Huge problem. We're continuing to train people wrong in this, and we're reporting all the wrong things about our statistics as well. Huge problems with our use of p-values and how we analyze evidence. Next slide. And we have these problems of publication bias. Could you just click on this, Matt, because I think there's a bit of text to come. Yeah, if you look at effects and the precision of the effects, you end up with this funnel with large studies at the top and small studies at the bottom, and they should fill the funnel because the small studies are just more variable, but they don't. The funnel isn't symmetrical. And this is telling you there's a difference between large and small studies, and it's one possible explanation for this difference is that the small negative studies aren't being published. So classic publication bias. So we have these publication biases. Next slide, Matt. And, you know, ultimately, a large part of this problem comes from the fact that our primary dissemination mechanism for information is the scientific paper. And it's worth remembering that the scientific paper was invented as a dissemination mechanism for a gentleman's club, which is what the Royal Society was when it started out. Now, that was fine back then when you wanted to sit with your smoking jacket, you know, but I'm concerned with trying to inform the biodiversity crisis with scientific data. I shouldn't be hampered in that effort by the fact that my dissemination mechanism is this totally outdated, ridiculous thing. Next slide. We have this problem, but we focus on novelty, not the strength of evidence. No, we're obsessed with novelty. So we don't think about what's the risk of bias of these studies that we've got, that we're combining. How good is the experimental study design? Is there any confounding? We can't assess the precision because all these different studies have measured the same construct on different scales that have got different statistical properties, so we can't combine them. We can't assess inconsistency properly, genuine heterogeneity between studies for the same reason. They've all measured the same construct in different ways. We've probably got publication bias, but we probably don't even have enough studies that have been measured in a similar enough way to be able to assess that. And then we're probably looking at outcomes that are surrogate outcomes that we're not really interested in as well. But the real problem is that because we don't adopt our fair data policies, you can't even tell what the strength of evidence is. Next slide, please, Matt. Okay, but there is hope. And I said maybe 10 years ago at a conservation conference, we should just chuck all of our studies in the bin. All that's really changed since then is that now I say we should really just chuck most of our studies in the bin and then set fire to it, because all they're really doing is obscuring what little evidence there is. But there is some hope that we can sort things out, and the hope is all of these open science practices that are the focus of this conference. Pre-registration is critical. It's the only way we can sort out publication bias. If you go back to that Salmonid paper where they measured all of those 15 outcomes, if they'd pre-registered, this is the one that matters and this is why it matters, then we will be able to tell whether or not they were just reporting the statistically significant results selectively. As it is, we have a very strong suspicion that that's the case, but we don't know. We need to spend much more time as ecologists arguing about what we should measure and how we should measure it, and then adopting that as a standard. It should be very difficult to get funding unless you measure that thing in that way. And this is the idea of core common outcomes, which are routinely used in medicine. Have the argument before you do the study, not afterwards. Open data, so that you're not sitting there looking at these graphs trying to figure out whether or not it's a standard error or a standard deviation or what the degrees of freedom are. Having to run little simulations just to reconstruct the data to find out what the variance is. It's a nonsense. Reporting standards, again, to facilitate that. More evidence synthesis. Partly because our primary studies are in such a state, over-reliance on primary studies is a real area of risk if we want to produce decent policy guidance. And then finally, the methods development. We need the meta-epidemiology to look at what's going on where and automating these processes as much as possible so that they align both with fair processes and make data synthesis more efficient. And that's pretty much the end of my rant. Thank you very much, folks.
Thank you. There has not yet been so many questions on Slack, but there might come some later that you may answer after a while. But I will just give you one question from myself while I have the chance. You mentioned that there is a problem that, for example, the funding agencies are too concerned about novelty always. But if you would have to pre-register to the funding agency what you're expecting to find and actually get money if you do follow up on what you say. Do you think it would go the other way around, that it would be only the very safe things we would end up studying because there's only so much we can predict in advance before we start our study?
Yeah, I think the pre-registration thing is just about avoiding the bias. So it's to stop you from spinning the sexy story to get the pathway to impact from the grant. So I think there is an issue there. And there are also issues of incoherence between what the objectives of the grant round actually are, or the call are, and how you shape your research. But I think sometimes what we need is we need very boring evidence that's kind of A-level geography type stuff, not some sexy new method on something that, sounds all very wonderful and is interdisciplinary in some exciting new way, but doesn't actually deliver anything. So sometimes boring building blocks are what's needed to construct that wall, rather than just shiny new things.
I see your point. Let's try to get a hold then of the last speaker.
We're having some trouble.
You're having some trouble, okay.
So we'll be a minute or two.
Yeah,
trying to sort that out.
a minute or two. Okay, then we have to figure out what to do a minute or two. Anyone has any good questions somewhere in Slack maybe that we could discuss? There is lots of discussions going on actually in the different threads I've seen, and tips being sent here and there, and looking promising here now that something is coming up on the screen at least. It's not so easy for the poor speakers here being tossed back and forth in the program all the time, never knowing when they're about to start speaking. But at least now we
can you hear me?
We can hear you, Cecilia, and we also
Yeah,
have
good.
your presentation on the screen. So this looks very promising.
And that's fantastic.
So,
yes. Good start.
Yeah, please start.
Yeah, shall I just go ahead?
Just go ahead,
Yeah.
please.
Yes. Okay. So the title of my talk is Open Scientific Knowledge Synthesis with experiences from the Norwegian Scientific Committee for Food and Environment. So I'd like you to draw attention to VKM. This is the short in Norwegian for the Norwegian Scientific Committee for Food and Environment. And this VKM is used through the presentation. So this presentation is sort of divided into three parts. It's first I will talk a bit about what is an open scientific knowledge synthesis. And then I will talk a little bit about VKM, introducing VKM. And then I will give some examples of VKM scientific assessments. But this talk is based to a large degree on a discussion that has been conducted by the scientists in VKM. And they have sort of done some reflection over the work that has been done in VKM, and they have written up this report that is shown here on this slide. And I'd like to mention the names of the people who has been in charge of this, and this has been Vigdis Vanvik, Edel Elvevold, Merete Åsmo Finne, Merete Hofshagen, Anders Nilsen, and Inger Lise Steffensen. And it's been a large discussion on this topic, and I think it's been very good and sort of timely to sort of discuss what's the position of our organization in the scientific world at the moment. So thank you for all that work that you have done with this. So open science, it sort of has started, it's sort of like a development that started in the 17th century, and how science is sort of used these days. The previous presenter had the slide here from the Royal Society and the community then, but science has changed a lot since then. We have now open science, and we have been discussing a lot in Norway, sort of open science and access to published material for everyone. But open science is so much more than that, and it's sort of illustrated in this figure here. It's lots of building blocks in open science. It's an open debate, open commissions, open publishing, open data, and open databases. It's open declaration of interest statements. It is, of course, declaration of interest is a very important thing to build loyalty and trust in the science that is made. Open peer review, open methods, open education, which is also very important, I think. Open review and knowledge synthesis, open authoring and open communication. So there are lots of things that has to be in place to sort of develop this wonderful world of the open science. So with the open science development, there has evolved this international organization that has been presented just recently as well, IPES. So that was presented here just now. That started in 2012. And IPCC that was established in 1988. And it's wonderful how scientists are able to sort of communicate out the science on this, and like on this climate issue. It's like everybody knows that we have a climate change, and they have been successful in communicating this to public and to governments. And of course, as you know, they got the Nobel Peace Prize in 2007. Another sort of international body that is of big importance for VKM's work is the European Food Safety Authority, EFSA. EFSA was established in 2002 as a result of the food scandals in the food industry in Europe. You might remember this mad cow's disease that happened in the 90s. And with EFSA, that's a body that ensures that there are regulation on food industry and also on agriculture. So it's from farm to fork. It's the whole value chain in food production. And it ensures that, like, for instance, pesticides and additives in the food industry, all these elements that can make food unsafe for the public. So it's EFSA is an important body in the EU system, since a society has to provide safe food for all its inhabitants. So it's an important part, especially for VKM as well. So about this conceptual understanding of knowledge synthesis, it has been discussed here a bit today already, but I'll explain this quite complicated figure here. It's knowledge production. That is, of course, all the science that is produced. Knowledge can also come from other sources of science, but I think science is the main focus of this conference. So we have single studies, single publication, but it's sort of into this knowledge synthesis where all the research that is conducted over a period, it's sort of reviewed and put together, and you draw out all the overall conclusion of the whole community, science community's knowledge at the moment. And by sort of scanning through all the literature that is available at the time, you will also identify knowledge gaps and uncertainties. So this knowledge organization can communicate back to the science community. You have to do more research on this field, and you have to get less uncertainty on this and that data and analysis on this. So there is a communication in this circle here with the scientists and the knowledge synthesis organizations, that where you have to focus your research. Then this knowledge synthesis go on to the policy formulation and management. And this is, of course, very important, and this is what we hope now with the climate research and the biological research that is conducted at the moment. And the communication back from the policy formulation and management can be on assignment of questions formulation, which is sort of a communication that will go backwards and forwards between these bodies, because the formulation of the question to be addressed is a very important part of the process of doing a good knowledge synthesis of a topic. When it comes to the policy formulation, it is of course other factors than the knowledge synthesis that is of importance for the policy formulation. And I think in this COVID situation that we have now, this has become very evident that it is not only science that controls the policy. This very complicated slide here is sort of explaining the whole process of the knowledge synthesis process. First, we have this assignment specification. Like I was saying here, you need to have a dialogue between the group who's going to conduct the synthesis and before it starts. So you have to have you get a first draft of the assignment, and this can be out on a public consultation, which is sort of ensures that the important part of the question is addressed. And having public consultation is sort of increasing the trust in the work that is done. So when then you have the sort of final assignment, and then you will start planning of the work. Then the question is, who is going to be the project leader? Who is going to participate of scientists in the project that is going to be done? What kind of competence is needed? Then you have to have a group that is going to approve the product, and you have to have a peer review scientist who can conduct peer review during the work. And then we are over in the next phase. This green phase here is when the actual scientist is conducting the assessment, which is also the longest process, of course, and they will have peer review and will evaluate drafts, and they will have comments, and the whole process that we know that is in doing this scientific work. Then at the end, it will sort of reach the commissioner and the stakeholder, and they will do an evaluation. Is the question addressed and answered in a proper way? And if it is so, and if it is approved, it will go on, and it will be developed some policies and regulation based on the scientific findings. This is this systematic review, step by step, formulation of question, search for relevant published literature, often in contact with the library, so that you can argue what is of good quality and what will be left in for the study, and what is of not so good quality and has to be left out of the study. Selection of relevant studies, extraction of relevant information, assessment of quality of the studies, data analysis of individual studies, maybe meta-analysis, and then you can answer the question. I'm sorry, I'm going the wrong way. So that was in short the first part of the talk, and I will then introduce VKM, the sort of Norwegian synthesis organization, who we are, what we work with, who we work for, and our role in the Norway's government and how we are organized. So we were established in 2004, very much on the model of EFSA. We have a main committee, and then we have 12 research committees on different topics under the main committee. And we are reporting to 4 ministries. We are reporting to Health and Care Services, Agriculture and Food, Trade, Industry and Fisheries, and Climate and Environment. So quite several topics that can be addressed. We are intersectorial, so which is a good thing in the work that we do. So we are providing these authorities with independent risk assessment and other scientific knowledge synthesis in the field of food safety, food production, and the environment. So we have 3 main responsibilities. We produce the scientific knowledge synthesis in the areas of food and environment, and our commissioners are the Norwegian Food Safety Authorities and the Norwegian Environment Agency. we are very happy to have these two commissioners because we can have intersectorial topics. We often see that a scientific question is better to be studied in the light of both food safety and environment issues together. This is sort of giving a better knowledge basis for the regulations when you have different sides of the topics studied, and it's also resource efficient to do it in this way. The second thing that we do, we are the focal point for the Norwegian in Norway for the European Food Safety Authorities, and we are Norway's scientific authority for CITES, which I will say a few words about more. So what do we deliver? We deliver risk assessments, benefits risk assessments, systematic reviews, and other scientific evaluation input. This is a photo from the European Food Safety Authorities. Here we have a scientific advisory forum. We sort of provide the 28 member states in EU participate in the advisory forum to give strategic research advice to the management board. Then we have a focal point, which it's doing the more administrative part and implementing everything in the national states. And then we have a communication expert network, and we have peer review network participation in EFSA, that is all the scientists that are participating in the expert panels. We are, VKM is also the scientific authority for CITES, which is the Convention on International Trade in Endangered Species of Wild Fauna and Flora. We have been appointed as a scientific authority by the Ministry of Climate and Environment, and we perform all scientific assessment necessary to enforce the agreement in Norway. We do only the science, and we leave the regulations to the authorities above us. We are not taking part in anything of the regulations. So what's our committee? We have almost 100 scientific members with a diverse background. So our committee have a wide interdisciplinary competence, and our members are employed in universities, colleges, and research institutes in Scandinavia. We also have a secretariat that is staffed by scientists. When we appoint scientific members to our committee, they of course have to have the relevant expertise to the topics that we are going to address. We have an open application process, and the candidates are reviewed by a Nordic assessment committee. So when the successful candidates are determined, then they are appointed for 4 years at a time by the Ministry of Health and Care Services. One very important thing in the work that we do is the impartiality at all levels. So everyone who performs work for VKM must evaluate their impartiality, and the declaration of the interest statement are published on the website. And impartiality is taken seriously by the scientific steering group, panels, and project groups. So minutes from meetings held by the scientific steering group and panels are published on the website. It's very important for us to always have an impartiality declarations since the findings are used for regulations. It's important for the trust in the results. VKN is independent. We cannot be politically influenced regarding scientific assessment and conclusions. VKM is unaffiliated with any institution, but the Secretariat is administered by the Public Institute of Health. The members of the Scientific Committee are appointed for their expertise and do not represent the employees. This is to make sure that there is no interest from the, for instance, university or research institute when it comes to funding or maybe scientific traditions. So they are sort of paid by the committee so that there is no loyalty trouble for the work that they do for us. VKM can also have make self-initiated risk assessment, and this also ensures that the scientists can do scientific work on an independent level. So they can do studies on an independent level. This is the secretariat. We are 23 people. We have 2 scientific managers, 16 project managers, 2 on communication and 2 on administration. And this secretariat is very highly educated. Most of them have a PhD, which is necessary because the risk assessment and other products that we produce are at a quite high level, and it's quite advanced work that is done in the committee. So these are the scientific groups that are present now in 2020. They have been like this for the last 4 periods. There is, of course, possible that some of the groups might be changed, that there will be added new groups if this is needed. But it's sort of a wide range of topics that can be treated in VKM. So who does what in the risk assessment? The scientific committee carries out the risk assessment, and then risk communication is done by us and the commissioners. But when it comes to risk management, that's done only by the authorities. This is not done at all by the scientists. So it's a very clear divide between risk management and risk assessment. I think I'll skip that one. Guidelines and methods development within the risk assessment. When we conduct a risk assessment, we start to work with establishing a protocol for the work that should be done. With this, we sort of decide what method to use, what doses to be evaluated, what kind of papers to use, and so forth. So the whole project is described in details before it starts. And this protocol is often sent out to an open hearing so that other scientists or interest parties can comment on the protocol. This is to build trust in the work that is done. Then we carry out a systematic literature research, and you have to sort of document what is left in and what is left out, and sort of have a reason for doing this. So systematic reviews of risk or benefit. Then we have an evaluation of quality of data, and we conduct statistic analysis and modeling of the data. The risk communication should be open and transparent, and all assignments are described on our web page. All finalized risk assessments are summarized and published in full on our website, and all risk assessments should be verifiable. So, last part of the talk, some example of VKM scientific assessments. This is an example of the wild boar population, and it's commissioned by the Norwegian Food Safety Authority together with the Norwegian Environment Agency. And with this, they did an estimation of the population growth in a changed climate and looked at the effects of the food safety, animal health, agricultural production, and Norwegian biodiversity. And based on this work, the Norwegian authorities has developed an action plan against wild boar for the period of 2020 to 2024. Another example of great importance is the risk assessment of radioactivity in food. And this was commissioned by the Norwegian Food Safety Authority, and this was to evaluate the current permitted maximum levels for radioactivity in foods and drinking water. And this was assessed to affect the conducted measures to reduce the amount of cesium-137 in foods after the Chernobyl accident. This assessment is on microplastic, and this is a self-initiated mandate from the steering committee of VKM. They wanted to summarize the state of knowledge on the presence of microplastic in the environment with the implication for the ecosystem, terrestrial and aquatic organism, food production and human health. And not surprisingly, they found that it was not adequate of data to make very clear conclusions. This is another example. It's commissioned by the Norwegian Environment Agency, and it's an assessment of the risk to Norwegian biodiversity from the import and keeping of terrestrial arachnids and insects. In this work, 6,000 species of insects and spiders was evaluated regarding Norwegian biodiversity. And the experience from this was that it was a very high number of species with 6,000, and there were identified lots of knowledge gap on this volume, of course. Then the final example now is from the CVD, the chronic wasting disease in Norway. This assessment was done in 2016, 17, and 18, and commissioned by the Norwegian Food Safety Authority and Norwegian Environment Agency. And the assessment here was on food safety and animal health, management strategies to prevent disease transmission. And in 2017, VKM concluded that culling of the Nordfjella reindeer herd was necessary, and this was also conducted. But unfortunately, we have now discovered a new case of CVD in Norway, and we have recently received a new assessment commission that will be conducted this year. Yes, thank you. So I think this was my presentation. So I hope that you find that you see that we have important work to do in the science community, and that we have a lot of important and interesting topics that should be addressed. And that is the science is very needed to give useful advice or not advice, but useful knowledge for the government to do good regulations. So thank you so much for your attention.
Thank you. I think we have, or take the time for one question. There's one question here at Slack. How would more open and fair data be beneficial for the VKM's assessments?
I think open data is essential for the trust in the evaluation that is done. So of course, and sharing of data as well. So it's absolutely essential to have open and shared data. So that would be a very big benefit for us. Yes.
And now there's one more. I take that one as well. In your description, VKM, very similar to IPBES in its working structure. So would you consider developing a data management policy for VKM?
Yeah, I think so. Definitely, and it's very interesting to see how other bodies sort of organize this. So it's very interesting to listen to the talks here on this. So this is something to look deeper into. Yes, absolutely.
Thank you. Then I would really like to thank all the three speakers of these sessions, particularly for being patient and surprised about speaking times and whether it would work or not. So thank you a lot for your flexibility. And then I think there's time for a summary as the final part of the day. Some final words or something.
Yeah. I think I would need to... Can I take that one? And now this is on, yeah? Perfect. Okay. Yeah, like that? Yeah. And let's see, I will do this and maybe... Yeah. Okay, we'll try like that. So Ingeborg said that I should make a summary of the day. I'm not going to do that. I think there are too many thoughts going around in my head, and I guess it's the same for the rest of you. I think this has been a great day. A lot of interesting discussions. I am also glad to see that there is a lot of discussions on Slack. we were quite curious to see how well that will work. I mean, we are quite used to sitting in a group room like this and discussing, but to see how well it works, to have the live discussions online going on at the same time, that was something that we were interested in, and I think it looks like it's been working well. So I will not conclude much from this day. I think the discussions will go on. I really found all the presentations really interesting, and I think they all cover different aspects of this bigger picture that we're trying to discuss in this colloquium. Couple of points. I mean, I had to write this down before the last session, so maybe I would have put some other additional points on the slide in addition if I had done this after the last session, which presented really interesting and slightly different viewpoints on the state of the situation, but they all agreed on the way forward. I think it's about opening up the research process, verifying the data, and improve reproducibility. I think most presenters has agreed that this is much about culture and increasing the know-how. It's a little bit about building infrastructure and technological stuff. Know-how. I think at least the second talk with Anders also pointed to the point, or not, but is the viewpoint. That should also mean using open community standards more than we do now to increase the reproducibility and the value of the data that we share. There has been some discussions I see on Slack about the and how we give credit to those that share the data. I think that's an important point, and I think that's to increasing the amount of data that is probably shared. Probably, it's late day. I'm not able to pronounce. Shared in according to best practice principles. And I think this ties into the discussions about how publishing high quality data, how that can be more valued in the research community as a valuable output in its own, and how this can also be valued more by the society as a standalone or a related output from the scientific process. Some practicalities for tomorrow. Later today, you will all receive an email with a link to the second part of the survey. Please fill in. You have received 3-4 mails, reminders and so on about the first part, but there are 3 parts to this survey, so you should all reply to this one. And you will get this mail later this afternoon, and it should be filled in before 9-ish, 10 tomorrow morning. And it's even shorter than the first. And it's shorter than, it's definitely, it is shorter. It's much shorter actually. But you have to open it to see if we are speaking the truth now. But we promise it's shorter. Tomorrow will be more interactive, so meet in time so that we can all organize the group works and in the breakout groups, both those that are present here and also the live or the online audience. Have a look at the last version of the program. It shouldn't have changed the last few days when it comes to timing of things, but please have a look just to be sure that you are on the right time. For those that are here and are joining the dinner, it will happen here at 6 o'clock, Norwegian time, obviously. With that, I would like to say thank you to all the presenters, the session chairs, and all the participants, both those that have participated in the discussions and those that have watched the live stream. And I would also like to thank those that are behind the scene, that have ensured that this has worked out really well so far. You don't see them because they are behind the scene, but it's Lasse, Juliette, Lars, Markus, Annette, and thank you all for the setting up and sorting out the technicalities today. So with that, I think we end the day and look forward to meeting you all again tomorrow. And no questions after this brief presentation.